Ana, može?
Može.
Dobro. Good morning, everybody. The Amazing Power of Fetal Brain was the title of previous webinar, which our academy organized 2 weeks ago, was very well attended. But this morning is a very big pleasure to see that 460 participants are requiring possibility to follow up us. Why amazing power of fetal brain? Just this figure to take home message. Our brain can perform 10 quadrillion operations per second without you knowing it. And one of the world's largest supercomputers takes 40 minutes to calculate a single second of human brain activity. That's why WHO proclaimed 21st century as the century of the mind. And in a recent publication, it has been illustrated that more than 35% of the overall cost and burden to the healthcare system of the European Union goes for investigation of the brain and peripheral structures. Most of the activities happen during fetal period, and the rate of 250,000 neurons per minute, neocortex is intensively under construction, and very impressive figure, 99% created during the fetal period of life, and most of the 100 billion neurons and related major pathways found in our adult brain have formed already in fetal period and are present already at birth, making our profession very, very responsible. And the window of opportunity for organization for brain development is obviously perinatal time. It is even easier to assess fetal neurology than neonatal, and we with new tests are more successful than neonatal assessment. And during fetal period, brain developed very rapidly, and understanding the course and timing of fetal neurodevelopmental events in correlation to both motor and sensory system development, and will be essential for determining how environmental effect can affect certain structures as well as function. This is very complicated thalamocortical architecture, but this is the key to understand neocortex as a whole and the emergence of intelligence, and to understand how cortical hierarchical levels interconnect. Sometimes difficult, but the biological basis of emotional and social behavior is formed by activity of complex local and remote neural networks and related basal ganglia. Ladies and gentlemen, last slide. Big question. Are we ready to investigate cognitive function of fetal brain? With all of my seriousness, I can say yes, we are. We are advanced. Zagreb Center and my group consisted of 12 people in Zagreb, does first studies, published the first study in an overview paper in Eberhard Merz edited journal. So cognitive function is the future, and the future already started. Very well inside the future are young Donald family, the largest educational issue in the world. And you can see here, apart from Senior, Saling and Maeda, you can see that our two leaders for today, Ritzi Kupu and Eberhard Merz, among many other creative people, are particularly advanced, particularly appreciated within Donald School, within Academy, within Board of Ultrasound, and we are very grateful to both of them. On behalf of International Academy of Perinatal Medicine, you see them both in the first row. This is a society now in 15 years. We'll have meeting in June, annual meeting in Marrakesh. and we are going to report there about research project which is under umbrella of academy, including wide spectrum of structural and functional development of fetal brain. Thank you all for contributing to this remarkable webinar on power of 3D ultrasound in advanced fetal neuroimaging. And thank you, Ritsuko, and thank you, Pooh, and to all speakers this morning. I'm sure participants will truly enjoy first-class information, attractive speakers, and you will find this webinar both stimulating and pleasurable. Welcome.
Thank you very much, Asim. And this is the webinar on power of 3D ultrasound in advanced fetal neuroimaging. So we would like to introduce the first speaker, Professor Shigehito Yamada from Japan, Kyoto, Japan. And he is a professor at Congenital Anomaly Research Center, Kyoto University. And he is originally, he is the OBGYN doctor. So he is still working as the OBGYN doctor and the professor at Congenital Anomaly Research Center in Kyoto University. So please, Professor Yamada.
Yes, can you hear me?
Yes.
Okay. So thank you for giving me a chance for my presentation on the title Neuroimaging. And this is my simple introduction. And I was medical student. When I was medical student, I start my research in Congenital Anomaly Research Center about human embryo. And 5 years, I worked as an OBGYN doctor as a slave. And the when I was a graduate student, probably this is the first meet with Dr. Fu, and as the postdoc fellow at the NIH and staff scientist and Congenital Anomaly Research Center. And now our current position is professor of human health science. This is a teacher, anatomy teacher, and also the manager of collection, and also the clinical geneticist in clinical genetic unit, Kyoto University Hospital. And probably this was the only participation in the IAPM conference. This is me, and not to conductor, but chorus conductor as a ceremony. Okay, this is my current job. So today's I have a lot of roles, and today's talk is about this area. And sorry, I skipped rehearsal last week, and today this is the agenda: normal development of human embryo and the Congenital Anomaly Research Center Kyoto correction and development of CNS. Okay, human embryo development classified by the Carnegie stages. This developmental stages reflects the morphological change of the embryos. And this, the first is the stage 8, 1 millimeter, 18 days. Days means the day after fertilization. It is 2 weeks smaller than gestational weeks. So the 4 weeks, 4 day means the gestational weeks. So the development starts a small plate of tissue. This is only 1 millimeter. Next, neural groove comes up and neural tube starts the development. And we can see somite. And the embryo grows up, upper extremity comes, and lower also do. And the primordial hand and foot, in these stages, they still have tail. In these stages, stage 17, 18, 19, I think they look like human. The previous stages, mouse embryo is also a similar shape, but in this 17, 18, 19, I think they look like human. And the finger starts to appear, the head is going up, and the hand and foot appear to be longer. This is Carnegie stage 23, 9 weeks, 3 days of gestation. Organ genesis has almost been completed. It means here is a very small human. Each organ is small and still immature, and it doesn't work. All the organ has been made by this stage. Okay, there are several human embryo large collection, and one is a Carnegie collection of human embryology in USA, and next is a very Schmidt collection of human embryos in Germany, and here is the Kyoto collection of human embryos. Okay, Kyoto collection of human embryos. This is the founder, Hideo Nishimura. Professor Hideo Nishimura started collecting embryos from obgyn doctors in 1961. And in 1975, the Congenital Research Center of Kyoto University Graduate School of Medicine was established for stable storage. Now the number of embryos comprises over 40,000, and various research and analysis continues till now. The characteristics of the Kyoto collection, most of the specimens were obtained by artificial abortion for sociological reasons under the maternal protection law of Japan. So it means they're legal collection, not illegal collection. Most of the embryos have been derived from healthy women within normal course of pregnancy in their first trimester. At the ending of obstetrics, this is not examine the aborted embryos. So it means the Kyoto collection can be considered to be representative of the total intraterritorial population in Japan. It means a ratio of normal abnormal in utero at each gestation weeks can be elucidated using this collection. The change of ratio can be observed in the longitudinal study. So the Kyoto collection, variable samples in Kyoto collection for epidemiological analysis, as well as morphological research, I think. And the classically, we use dilatation and curettage manner for the main technique for artificial abortion. Recently, we use suction curettage has been used. So it is impossible to collect these large number and damaged embryos and the embryo collection. Okay, here is our center, and when the COVID-19 issue is over, please visit us. And this is microscopic room, and we have a lot of sets of serial sections, and 547 cases normal embryo, almost 500 cases with abnormal external appearance. And using these precious sets of specimens, I published paper about whole prosencephaly embryo. And here is stock room. So here are the human embryo samples, and this hand is my left hand, so probably it is normal size. So this is a 5mm tube, a 10mm tube, 20mm tube for MRI scan, and this is almost 6 weeks, 7 week, 9 week embryos. Yeah, but these embryos we call famous embryo because they are used for many textbooks all over the world. So here is an example. Now here's a famous textbook of human embryology, and you can see the photos of Kyoto embryos. Okay, this is a summary of the development by 9 weeks of gestation. Major developmental events occur and finish. And here is a movie of the human embryo development, but we don't have enough time, so I'd like to skip the movie here and here and here and here. Very rapid development. Okay. So this is the today's main theme of the is the development of CNS. Yeah, here's the textbook, and closure of the neural tube is the start of the development of the central nervous system. And this means formation of primary neural tube. As you know, there are primary and secondary neural tube. The primary neural tube formed by fusion bilateral neural fold and become up to the lumbar spinal cord, although there are some differences depending on the literature. The secondary neural tube is the mesodermal mesenchymal cells aggregate in the tail bud, And the body grows caudally due to growth of tail buds. The center of aggregated cell mass becomes a ring. This is canalization and forms the secondary neural tube. And fusion of primary and secondary neural tube makes a single neural tube. Yeah, a lot of diseases and syndromes are derived from secondary neural tubes, so we have to know it. And here is a list of the diseases. And here are some terms about the neural tube. And it's a little bit confusing. And the primary and the secondary neural tube, I said primary neural tube formed by closure of neural tube, secondary neural tube formed by aggregation of mesenchymal cells. And the other was said is the anterior and posterior neural tube. Anterior neural tube becomes the brain. Posterior neural tube becomes the spinal cord. So here is a scheme of the primary, secondary, and anterior, posterior. And the PNT means the posterior neural tube, and N1 and N2 mean primary neural tube and secondary neural tube. This is my very old paper, almost 17 years ago. And the first author Saitsu, it's here, Professor Saitsu. And I've associated the tail buds with human tail. So here's a paper about classification of the human tail published last year. So please check it if you're interested. And this is very basic knowledge. Brain segment is primary and secondary brain vesicle. And here is a primary brain vesicle consists of prosencephalon, mesencephalon, lumbencephalon, and secondary brain vesicle, telencephalon, diencephalon, mesencephalon, metencephalon, meroencephalon, and finally the brain is made from hemisphere, diencephalon, mesencephalon, cerebellum, pons, and medulla oblongata. And my talk, next short talk is about the development of telencephalon. Yeah, encephalization is the characteristics of the human, and you can see here. Okay, here. Here is a human brain, and the other is the other not human brain. And the development of the human brain occurs during the fetal period and also during postnatal time. And here CRL 23.4 millimeter is the end of embryonic period, almost 12 weeks of gestation. And at this stage, the cerebrum is still small and the midbrain is completely visible. And oh, sorry, does it work? Nope. Oops. So, okay. And here is a movie of the our 3D reconstruction. Can you see it? And probably this is main event of my presentation today. So please give me almost 3 minutes to show development of a human brain. And this is very early stage, candidate stage 13. And this is only prosencephalon, mesencephalon, and lumbencephalon. And next, candidate stage 15, we can see yellowish, whitish telencephalon, very, very early telencephalon is here. And next candidate stage 19, we can see two lateral ventricles, but the telencephalon is still small. This is the end of embryonic period, candidate stage 23. This is the sagittal section of candidate stage 23. And here is the fetal stage, 86 millimeter fetus. Yeah, the cerebrum is become very larger. And here is the 170 millimeter, and but we can see insular cortex, because the development of cerebrum is not completed. And here is the almost 20 or 22 weeks of gestation. We can see a big sulcus or something. And here is a such a section. I love this movement, and very older 3D brain synchronized. Okay. And the final few minutes, I'd like to talk about the colloid plexus development. And here is a colloid plexus development. Colloid plexus has developed after neural tube closure. The order of appearance is fourth ventricle, and next lateral ventricle, and third ventricle. Colloid plexus of third ventricle complete its differentiation earliest among these three colloid plexus, although it appears last. It is easy to find, as you know, it is easy to find by fetal ultrasound until middle of fetal period, because it is relatively large. And do you know the developmental staging of colloid plexus? Here is so, and if you know, you don't have to concentrate in this section. There are 4 stages depending on histological observation. Stage 1, 2, 3, 4, and 7 week, 9 week, 70th week, and 20th week after fertilization. Sorry, after fertilization. And here is our research embryonic colloid plexus, 6 week after fertilization, 7th week, 8th week, like this. And colloid plexus appears at 7th week after fertilization, as we see in the last table. And it gets larger in only a week. And here is a chart about histological observation, our samples. And result of histological observation, the Carnegie stage 22 and 23 is a border of the colloid plexus stage 1 and 2. So stage 1 and 2 are showing embryonic period. So based on our observation, probably this table should be changed like this red characters, 1 week or 8 week or something. Okay, the last 1 minute, I exhibit very fast congenital anomalies in Kyoto collection. External anomalies have been diagnosed under light microscope. And here is cleft lip here. And this is also cleft lip. Polydactyly or left upper lip. This is oligodactyly. Probably you are interested. This is exencephaly, open neural tube. And this is the next step of exencephaly, Carnegie stage 20. You can see brain. And this is severe holoprosencephaly, and this is almost cyclopia. And my very old paper, I wrote about this cyclopia. And this is very beautiful cyclopia. This is very complicated, severe case. Okay, this is summary. Several human embryo collections in the world were introduced with emphasis on Kyoto collection. Normal human development has been overviewed with a focus on neuroembryology. And finally, very, very rapid, the photos of human embryos with external malformation were exhibited. Thank you for your attention.
Thank you very much, Professor Yamada. That was a very, very excellent and wonderful lecture on the human embryos. Thank you very much. So we will have the discussion at the last moment, after all of the lectures. So please write down your questions and comments in Q&A box. So Eberhard, would you please introduce the next presenter? Eberhard? Eberhard, are you sleeping?
Hello? Hello.
Hello.
Can you hear me now?
Yes.
Yes, okay. First of all, it gives me great pleasure to welcome you all to our 3D webinar, Advanced Fetal Neuroimaging. And we had a very nice start. And now the next speaker is Reinhard Altmann from Linz, Austria. Dr. Altmann had his medical education at the Medical University of Innsbruck, Austria. Since 2009, he is senior physician at the Institute of Perinatal Medicine, University Hospital Linz, Austria. His main interest is perinatal diagnosis, and in particular 3D ultrasound of the fetal brain in the first trimester. His first paper on the development of the fetal brain was published in 2016. He is a very active member in the Austrian Society of Ultrasound, and it's now my pleasure to ask Dr. Altmann to present his topic, sonoanatomy of the brain in the first and second trimesters.
Hello and good morning from Austria. Thank you for the possibility to present my ultrasound slides. It's very grateful to follow now with the ultrasound presentation of what we saw before in embryology. So let me start. My topic is sonoanatomy of the brain, the first and the second trimester. So I will start as we just did before, start with week 6 till week 11. And this is what we usually do when we diagnose a pregnancy at the gynecologist everywhere. This is the normal picture we get at a CRL of 5.1 millimeters. This is just for the introduction. This is what you can get when you do 3D ultrasound and try to get a little bit more information at the same time. You can see the amniotic cavity, you can see the chorionic cavity, you can see the stomodeum, and you find neuroporus anterior, as we just talked about, and you find neuroporus posterior. Then if you change to the 3D mode, you can even present this fetus in 3D as it was done before by Dr. Poole in 2012. And you can see the neural tube even at the CRL of 6 millimeters. And this is what we heard before, the closure of neuroporus. For us gynecologists in gestational week 6, the closure of neuroporus anterior, gestational week 5+3, and neuroporus posterior, gestational week 5+5. Now let me go a little bit further. This is the next fetus showing a CRL of 10 millimeters. And the first paper concerning that early gestational week was done by Blas in 1995. The pictures you can see in that paper are quite different from the pictures we can see here now. I'm going to show you. Very prominent in the middle of the fetus, you see the heart. And here you see the vesicles developing to the ventricles later on, starting with a rhombencephalic ventricle, mesencephalic ventricle, and diencephalic ventricle. This is a transparent mode already done by Rolnik in 2020, a very early gestation showing CRL of 12 millimeters. This is rhombencephalic ventricle, mesencephalic ventricle, and diencephalic ventricle. This is just before the diencephalic ventricle starts to migrate to the two vesicles, then growing to do the telencephalon. So this is the same fetus later on at the CRL of 15 millimeters. Once again, the cephalic flexure in the middle. You see the ventricles. And now diencephalic ventricle migrates forward and produces telencephalon impar. Telencephalon impar is the middle part in the development just before the vesicles start to migrate laterally and posterior. This is the actual view showing now telencephalon impar in the middle and the cerebral vesicle laterally in the front of the developing brain. And you see foramen of Monro in the front of the brain before it later on, after migration of the ventricle, comes to a position in the middle of the brain. This is the schematic presentation of the developing lateral ventricles and the ventricles as a complete development. On the left side, you see the fetus as it is in the normal 3D position, and then you can stretch that for the schematic presentation as we know it from the drawings from embryology. You see rhombencephalon, mesencephalon, and diencephalon, as we said before, now stretched. And this is the schematic presentation as well known. We just saw it without drawings before. Here you see the developing cerebral vesicle. This is a transparent mode 3D animation showing the ventricles as well. You see mesencephalic ventricle, diencephalic ventricle. You find telencephalon impar in the middle, and you find the telencephalic vesicle starting to grow and then growing back. You see the whole fetus, you find the hand as we talked before, you see the umbilical cord very prominent in the middle, you see the yolk sac on the left down, and this is a fetus at the CRL of 15.8 millimeters. This is the inversion rendering figure that was already done by Mitsu Kim and Timo Trich in 2007 and 2008 to show the developing ventricles. Now let's go to fetus at the CRL of 225 millimeters, a little bit older again, and presentation about the early developing brain was done in 2012 by Dr. Pu. And I want to show you in this picture the developing plexus choroideus. Plexus choroideus starts to grow in the front, starting with foramen of Monroe. The lateral ventricles start, and then communication between the ventricles over the third ventricle occurs. And this is something you can see very well on ultrasound in 25 millimeters of CRL. This is the plexus choroideus of the fourth ventricle. This is the same fetus in a little bit bigger. Here you see medulla oblongata and pons well visible in 25 millimeters. Now let me change to our normal topic, first trimester screening. This is our main object of investigation now. This is, on the left, is a slide from Professor Hochstetter, I received from the University of Vienna. And on the right side, you see an ultrasound picture of fetus at the CRL of 54 millimeters, corresponding to the histological slide. This is to present the ventricles. Here you see the diencephalic ventricle red, yellow the mesencephalic ventricle, and blue the rhombencephalic ventricle. The slide is to show the development of the vermis in the very early week of gestation. You find a CRL of 54 meters. On the left, in the slide from Vienna, and on the right, an ultrasound picture from the first presentation, from the first paper I did. Here you see medulla oblongata and pons. You see the vermis in that CRL of 50 millimeters complete. This is the next picture done with a newer ultrasound machine. You see the technical equipment is getting better and better. And here you see the details of the fossa posterior, medulla oblongata, and pons. You see the vermis, and you find plexus choroideus, and you find the tiny membrane of Blake's metapore, and below Blake's pouch, the future cisterna magna. Here you find an earlier fetus. On the right, you have a fetus at a CRL of 47 millimeters, and there is something different from the slides I showed before. You see what I presented before, the vermis and plexus choroideus, and there is Blake's metapore, as we saw before, and this is different. This is the membrane where the vermis is growing along, and this is anterior membranous area. So this is a comparison of the two CRL. On the left side, you find a CRL of 45 millimeters. On the right side, a CRL of 84 millimeters. And just for the orientation, the small vermis is on the left, the bigger vermis on the right, and plexus choroideus for the orientation, and that's the difference. Anterior membranous area on the left side. On the right side, anterior membranous area has vanished because the vermis has grown along. And you see a different angle of medulla and pons on the left and on the right side. The fetus in that week stretches. Yes. here we are. Now, the next is the presentation of the structures of the midline of just for the orientation as we had before, a mesencephalic ventricle in the midline. We see the entrance of aqueduct. Sorry. You see hypophysis here and chiasma opticum in that part. You see the roof of the diencephalic ventricle here, and this is the adhesio of thalami. The thalami do not close as they do later on. This shows a 3D animation, no, a 3D in all three planes. You see plexus choroideus of the third ventricle in the middle for the orientation. This is the plane, and here you see the lateral ventricles. You see foramen of Monro and choroid plexus of the third ventricle, as said before. And this is the histological slide in comparison. This is at a CRL of 78 millimeters. This is a slide showing the normal figure. The normal figure, as we call it, the butterfly sign. And the butterfly sign was the topic of many studies in the last years. The thing was that everybody tried to correlate the butterfly sign. I mean, the length of the plexus choroideus to different pathologic entities. Here you see the cerebral wall. The cerebral wall is still very small from the development of the vesicles. And you see the different correlations that were done. You see the correlation that Loureiro did concerning aneuploidies, and Manigault-Brauer did a correlation concerning ventriculomegaly. And last but not least, Professor Chow did a correlation to the open spina bifida. Nevertheless, it can just be an indirect sign because migration of the ganglionic eminence did not occur so far. So this is just for indirect, and we afterwards have to prove what's going on. So there are many papers concerning length, area, circumference, dealing with that differentiation between the choroid plexus and the length of the ventricle. On the other hand is the ventricle size. And one of the reasons why we cannot do directly deal with that is that the cerebral wall is very small, and the migration just starts in that week, starting with ganglionic eminence. You see in the ultrasound slide in red, ganglionic eminence on that slide on the right side is from histology, from Hochstetter, from Vienna, and this is a coronal view showing the migration along the wall. And here you see that it grows along, developing the later on presentable migrational figures. This is just to show the embryology. Once again, here you see the development of medulla oblongata and pons. This is pituitary gland. You see chiasma opticum. You see choroid plexus of the third ventricle. You see mesencephalic ventricle, vermis, and plexus coronarius. You see a commissural plate in the front and the roof of the third ventricle. This was done by Hochstetter in 1929 first, and this is a fetus with a CRL of 77 millimeters. Now let me come to week 14 till 17, where just two new papers came up. This is a slide showing the orientation. Once again, you see choroid plexus as a landmark in the middle, and you see choroid plexus of the lateral ventricles on the right side. You see it in the mid-sagittal view on the left side, and you see foramen of Monro, and this is a week 15 plus 3. And this is presenting the new data of Birnbaum. One was just accepted and the other one came out in 2020. And here you see corpus callosum, and this is carbon valley interposity presented by Birnbaum 2020. This is the next slide I wanted to show you. The next slide shows 16 plus 2 week of gestation. Here you see once again the corpus callosum, and on that part you see the crossing fibers, hypoechoic, crossing the midline. Now let me come to the week 18 till 25. 25 is the end of the second trimester, and week 18 is that week where the papers until 2 years ago were done. So this is the normal corpus callosum, and where all the investigators did their studies. That starts with Acheron 2001, goes further to Cullen in 2008, and many scientists worked on that topic. So I just took that part in week 18 to show how this early week presents the figures. Corpus callosum starts with rostrum, geno, and corpus being presentable in that week. There is a very early paper dealing with fornix, but there's still some work to be done to present the fornix. This is Cavum septipellucidi. This is the vermis, and the vermis shows increasing lobulation, and the brainstem vermis angle can be measured in this week as well. This is the next week. We have now changed to week 18 plus 6. And what's different from the week before, in this week, the splenium of corpus callosum starts to grow backward. This was presented by Rebaud in 2010. Now, this is week 18 plus 6, and here you see the migrational zones, the transient cerebral lamination patterns as presented by Pugash and Dr. Pu. Subplate zone, intermediate zone, and the ventricular or geminal matrix zone. And if you look at marker dot in the middle, you find the crossing fibers of the corpus callosum going a little farther up in the ventricles. This is week 22, and you see that the corpus callosum is growing a little bit further up. You see corpus callosum, you see cavum septi pellucidi, and the third ventricle. You see the anterior horns on both sides, and you see septum pellucidum, closum cavum septi pellucidi. This was done by and now just let me stop. This is not the end of that that slide. Here you see the Sylvian fissure, and the Sylvian fissure is an indicator for migrational disorders as well as the migrational zones in the last slide. And what I wanted to say concerning the papers of Professor Pu was the angle, of course, that she presented in 2019 as an indicator for migrational disorders. Just to change to the actual view is something everybody uses, and quite a lot of papers were done concerning this view. This is for the correct position, so 3D can be used for the correct position, a horizontal position, not to be oblique and to do incorrect measurements. You can also present a straight midline. In some migrational disorders, you can also find deviation of the midline. So this is also a necessary tool to diagnose the cerebrum. This is now the middle of the brain. You see corpus callosum in a fine presentation. You see the different parts of corpus callosum. You find the rostrum and the geno and the corpus and the splenium as it develops. Cavum septi pellucidi, and this is now week 25. One of the last presentations, publications that were done was on 2015 by Professor Paschai. And the same picture now showing the colliculus superior and inferior and the aqueduct. Vinyalis did a paper 2017 concerning aqueduct and aqueductal stenosis. This is the wedge angle that was done in the paper 2018 by Birnbaum concerning aqueductal stenosis. This is the normal variation of cavum verge. I wanted to show you. I'm sorry for the late week of gestation, but it was the best slide I had. It shows very clearly that cavum verge is a posterior extension backward of cavum septi pellucidi, a normal variation not associated with any problems to the fetus. Cavum septi pellucidi was a topic of investigation for many years, and Shaw did one of the first publications in 1969. Here you see cavum septi pellucidi, and backward of the columns of fornix overlying chorial plexus of the third ventricle, you find cavum verge. Here you see the posterior fossa in nice slides. This was done by Robinson 2007. You find fissura prima and velum medullare superiorus and inferiorus and the fastigium. This is in fetus in week 23. And this is just to present what in best conditions of ultrasound is possible, the different lobuli of the cerebral vermis. Thank you for your attention.
So thank you very much for this excellent presentation and for the nice pictures. Now, Ritsuko, who will announce the next speaker?
Okay. The next speaker is Karina Harat from Tel Aviv, Israel. And she's now in Israel, and she was from Brazil in 2012. And today she works as a physician at the ultrasound unit in OBGYN at Tel Aviv Medical Center. And also, you know, that she's working at Fetal Neurology Clinic in US. And she's now working with Professor Gustav Maringel. And Dr. Karina Harit is a promising young rising star on fetal brain imaging in Israel as well as internationally. So Karina, would you please start your lecture?
Yes, let's checking if you are hearing. Yes. Okay, I'll share my slides. Thank you very much, Risuko, for the invitation and for your kind words and for all the academy to having me. And we are speaking a little bit about midbrain abnormalities. So we will start speaking a little about midbrain anatomy, the genetics and the developmental stages of the midbrain, the clinical importance and the diagnostic challenge of these abnormalities. And finally, we will discuss some of the neuroimaging features of fetal anomalies affecting the midbrain using 3D dedicated neurosonography and learning some steps from MRI. Yes?
Okay. We don't see your slides, Karina. Please share your screen.
So Anna, you have.
Okay, just a moment. I'm sorry. So let's see if now you can see my presentation.
Good.
Yes?
Yes. Yes. Thank you.
I'm sorry. So I will pass through the outline again. Okay, and these are the topics that we are going to discuss today. So let's start speaking a little bit about brainstem. It's a big crossroad and communication center between the supratentorium and the infratentorium, the spine and the peripheral central nervous system. This is very, very important. A point of communication. The arousal of 10 of the 12 cranial pairs come from the brainstem, 2 of them arising from the midbrain, and we'll speak about it in a while. And the main divisions of the brainstem is the midbrain, the pons, and the medulla, and we are going to focus on the midbrain structure. A part of the strict relation with the brainstem structures, also the relation with the diencephalic structures is very, very important. Here we see the cerebral peduncles and the interpeduncular fossa and its relation with the chiasma and the mammillary bodies and the other diencephalic structures. The arousal of the oculomotor nerve, 3rd pair and 4th pair trochlear nerve at the level of the midbrain will explain why do we have ocular abnormalities and sometimes very severe ones whenever we have midbrain anomalies. So let's turn our brainstem, and here I thank my dear brother with graphic designer for this presentation. And we can see also the pineal body and its relation with the tectum, the quadrigeminal body. here another aspect, another view of the cerebral peduncles, and here through the cerebral peduncles come the motor tracts from the cortex, straight from the cortex, and the basal ganglia as well. The tegmentum lies with the nuclei and also many tracts that come from the supratentorium and the cerebellum. And an important area of the tegmentum is the cerebral aqueduct with the surrounding periaqueductal gray matter. The nuclei of the cranial nerves third and fourth stays and lie in this periaqueductal gray area anteriorly to the aqueduct. Finally, we have the tectum, the quadrigeminal plate or corpora quadrigemina, due to the two superior colliculi which are responsible for vision, and the two, inferior colliculi responsible for audition. And if we take out the cerebellum, we have another very important structure, which are the superior cerebellar peduncles that attach superiorly the cerebellum to the brainstem. The other peduncles will arise from the pons. And unfortunately, we really need to understand, we are obstetricians mostly, but we really need to understand embryology and genetics in order to understand the brainstem abnormalities. The process that involve the brain development involve first the anterior-posterior and dorsoventral patterning, which were perfectly, brilliantly explained during the lecture on the first trimester, but not also these very early patterning processes, but other processes will lead into normal or abnormal development of the brainstem and the midbrain in special. So the morphogenesis of the brainstem and all the Joubert syndrome and Meckel-Gruber and ciliopathy genes are very important in the development of the normal morphology of the midbrain. And another very important process also is the axon guidance. Many of those genes that regulate those processes are already known. And speaking about axonal guidance, this is a mostly new term for us gynecologists. We speak a lot about a migration of the neurons, but what happens with the body of these neurons, the axons, their growth, that pathfinding, this is very important, especially when we speak about the brainstem. Again, many of the regulatory genes are known, and mutations will lead to different patterns that we can use DTI and tractography to see the pathways, but we can use also ultrasound to see the morphological abnormalities. And why I am speaking about axonal guidance and speaking about midbrain abnormalities, because many of the anomalies will present morphological anomalies of the midbrain, L1 syndrome, Joubert syndrome and related disorders, tubulinopathies, which are part of lissencephaly, the encephalic mesencephalic junction dysplasias, and others. Some of them cannot be diagnosed by imaging. Some of them will affect other structures more than the midbrain, remembering that the corpus callosum is one of the most important structure and will be affected in all these abnormalities together with the brain structure. So what happens with those children? As you now understand what is the midbrain, so they will have motor problems because the motor tracts will be affected. They have abnormal eye movements as well. They will have hearing loss and abnormalities in the very basic functions, which is also another function of the brainstem, regulation of breathing, heartbeats, temperature, and others, as well as cognitive abnormalities. So for us that lead with a fetal imaging, it's a really diagnostic challenge. The brainstem anatomy is less known than other features of the brain. There is need of advanced neurosonographic techniques. We are not also aware not only with the anatomy, but also with the entities that we are dealing with. We have emerging overlapping, and some diseases will have only late appearance. So I hope this lecture will make a little order in this mess. We'll start speaking about technique of ultrasound. This is very important. Not only we need to use 3D and to obtain good images transvaginal whenever possible, but if we don't insonate the brainstem in the right angle, we won't get the information we want. So we will start through the posterior fontanelle 90 degrees with the brainstem structure, doesn't matter the gestational age, and we will be able to see the tectum, the aqueduct all the way through, and the tegmentum, the interpeduncular fossa, and the other structures as well. The problem with this insonation that there is something that we cannot tell, whether the diencephalic structures and the mesencephalic structures are really separated. And this is important for the mesencephalic diencephalic junction dysplasias. So we need to make a central insonation. And here 90 degrees with the diencephalon, we will be able to see the fluid in the floor of the third ventricle, depicting the massa intermedia, the interthalamic adhesion, and differentiating it from the roof of the mesencephalon. And this is very important to check on the cleavage and the normal development of the structures. Another very easy, but we are not used to do it, easy way to check on the midbrain is axial view. The same plane as we do the BPD, we go a little lower to the base of the cranium, and we see here very nicely our midbrain. I call it the Mickey Mouse sign. And we see also the presence of the fluid all the way through the aqueduct. It doesn't matter which gestational age it is. Tegmentum is a little more echogenic. Cerebral peduncles are a little more echogenic. Tegmentum is hypo as well as the tectum. And we see here the aqueduct very, very nicely. So here we have our Mickey Mouse. The interpeduncular fossa has an angle of about 90 degrees. We have no projections because the superior cerebellar peduncles are normally, they are very, very thin, and we barely see it on ultrasound. We can see it, but we barely see them. So we shall see no projection from the posterior aspect of the tectum, and the aqueduct is also seen very nicely. So there is a very important structural change that occurs between the end of the first trimester and the beginning of the second trimester that changes it all. And this is the reason that here in Israel, we do the early anatomy scan, not together with the nuchal translucency, but 2 weeks later on, because this transformation in the brain is very important to determine abnormalities as well of the ganglionic eminence migration that was brilliant exposed in the previous lecture. So here we see that in the end of the first trimester, the midbrain, the tegmentum is very thick, the tectum is large and thick as well, and the aqueduct is large, and this transforms abruptly to the second trimester. The midbrain becomes smaller relatively and comes to the middle of the brain. The other structures of the brainstem grow throughout gestation, and the vermis is rudimentary with a large choroid plexus, and we have inversion of these proportions, and the open fourth ventricle that closes after the development of the inferior vermis by 18 weeks. So 3D ultrasound is very important, especially multiplanar imaging and the use of the dot to be sure that we are located in the midline. Midbrain is very big, again, in the end of the first trimester, a little posteriorly located, not kissing the occipital bone. We are not speaking about spina bifida. And the brainstem is Z-shaped. What happens next? First thing, between 13 and 14, 15 weeks, so there is a week and a half lap, and the brainstem is straight. This is very important. The tectum remains large and the aqueduct remains large, but the brainstem goes straight in about 10 days. Superior vermis appears, and the fourth ventricle, the choroid plexus of the fourth ventricle becomes smaller until the full-blown first half of gestation picture, where the midbrain is small. We see nicely the close angle of the interpeduncular fossa, and the tectum is small. So this picture is very important, and you have to see it in mind. I rather, when I do early anatomy scan, to see this fetus than this fetus, because I need to check on this process. This is very important. We can check on it also in axial views and see the presence of fluid in the aqueduct all the way through gestation. And that's why during the last 6 or 7 years, we have developed the International Consortium of Fetal Midbrain and Hindbrain Malformations with some publications from our group. Professor Lebovich published nomograms using 3D ultrasound for the midline structures. And now Dr. Birnbaum he has his work in press with the early aspect of the brainstem in early second trimester, including also axial views, and we hope it will be available soon for you. This is another publication, a review article with features for the diagnosis of brainstem abnormalities. So I will first speak a little bit about morphology, and then we will enter diagnosis. So what are the main abnormal midbrain findings? We won't have time to speak about all entities, but I think this list is important for you to make order in your mind. First of all, aqueductal stenosis, and the most severe form, mesencephalosynapsis, so fusion of all the mesencephalic structures like a bulky structure. Brainstem kinking, the persistence of the first trimester. A pattern, the molar tooth dysplasias, we speak on the pathophysiology of the molar tooth, hypoplasia and severe hypoplasia, or thick and enlarged tectal plate, deep interpeduncular fossa, the asymmetry. This is very tricky finding. We need to check whether one side and the other are just alike. This is another use for 3D multiplanar imaging. The diencephalic mesencephalic junction abnormalities, the presence of heterotopic tissue, either from gray matter or from white matter in the interpeduncular fossa, so masses in the interpeduncular fossa, and abnormal dimensions. Unfortunately, we have no time, but this is a list. You need to check on the presence and absence of every finding like this. Speaking about kinked brainstem, Walker-Warburg and alpha-distal glycanopathies are the main representatives. So whenever we see a Z-shaped brainstem after 14 weeks, immediately we need to rule out alpha-distal glycanopathies, and Walker-Warburg is the most severe form. But other lissencephalies, like the tubulinopathies, especially TUB-A, which course with severe lissencephaly, and lissencephaly with cerebellar hypoplasias, but also other abnormalities, they course with a Z shape and sometimes very, very severe brainstem. A common finding of everybody is ventriculomegaly, because many times these abnormalities at the level of the midbrain will lead to aqueductal stenosis. So the presence of absence of ventriculomegaly doesn't help, but the very early presentation of ventriculomegaly really makes us suspect on Walker-Warburg, because their presentation is much more severe and early than other presentations like tubulinopathies, L1 syndrome, DMJ dysplasias, and Mobius syndrome. And later on, the presence of lissencephaly, of course, will help us. So how can we diagnose aqueductal stenosis even in early pregnancy? As a matter of fact, the earliest, the easiest, because the large aqueduct is very easy to depict. Whenever we see no fluid in the aqueduct, fusion of the structures, we are facing aqueductal stenosis. But there is something very, very important. Do not forget to insonate 90 degrees fashion to the aqueduct. If you insonate 0 degrees, so through the sagittal suture, you cannot tell nothing about the aqueduct and not to be sure whether it's open or not. But here we have a very nice posterior insonation, no fluid at the level of the aqueduct. I forgot what I am looking for. I go to my reference. So this is what I am expecting. Okay, no fluid in the aqueduct. We are facing aqueductal stenosis at 16 weeks. First thing we do, check on the brainstem. Is it Z-shaped? No, here the brainstem is not Z-shaped. So although dystroglycanopathies may course with normal straight brainstem, but probably Walker-Warburg is not here, then we go to the cerebellum to check for the presence of rhombencephalosynapses, and we check whether this fetus is male and he has adducted thumbs to check on the presence of L1 syndrome. So this needs to be automatic in our brain. Speaking about L1 syndrome, speaking about L1 syndrome, so we have hydrocephalus with or without aqueductal stenosis. Although aqueductal stenosis is present in 80% of the fetuses, always corpus callosum abnormalities, but they will appear later on, and variable degrees of cerebellar and brainstem hypoplasia, normally less severe than the dystroglycanopathies. And if we do DTI, we will check the absence of the pyramidal tracts, and they are quite pathognomonic, but DTI is not available to most of us. So this very new review for L1 syndrome divide the syndrome in three aspects of the spectrum. Hydrocephalus with aqueductal stenosis is the most severe one with the most severe intellectual disabilities. Male fetuses, S-linked recessive pattern of inheritance, hydrocephalus, abnormal corpus callosum. But remembering the differential diagnosis, most individuals with L1 syndrome have no abnormality in other systems but the brain. An exception may be done to Hirschsprung and abnormal bowel findings, but if they appear very later on in pregnancy. And I am really sad still to read in the medical literature that they think the diagnosis must be postponed to 24 weeks. I think whenever, as I said, we see early hydrocephalus in a male fetus, adductor thumbs is very prevalent, go and check for the mutations. As I told you, L1 syndrome is less severe than alpha dystroglycanopathies, and sometimes we will check on these fetuses only later on in pregnancy. Mild ventriculomegaly 14 and 12, very irregular ventricular walls, abnormal sulcation pattern. If we check on the cerebellum, cerebellum is quite okay, 32 centile, no communication between fourth ventricle and cisterna magna. So you see, these are quite easier features. We are not going very advanced. But whenever we see that, then we need to go a step further, and we see no fluid in the aqueduct. These fetus undergo MRI. Of course, the ultrasound made depict the corpus callosum, in my opinion, even better, the boundaries that the MR, but here I only have the MRI to show you. And we see a thin corpus callosum, not sure about the normality of the splenium. The brainstem is not hypoplastic, but the midbrain is hypoplastic. No fluid in the aqueduct, thick tectum, thin tegmentum. And this is a fetus with L1 syndrome. This is a very nice paper that took 138 fetuses, candidates to L1 syndrome, boys, with hydrocephalus, and then checked for 5 features: hydrocephalus, adducted thumbs, corpus callosum abnormalities, stenosis of the aqueduct, and because this is a neuropathological feature study, they checked for corticospinal abnormalities. The most features most likely to have X-linked hydrocephalus. The next group is the L1 chem-like. They have all features, but you go to genetics, you don't find any mutation. And then we have the less features belonging to the aqueductal atresia spectrum and other syndromes that concur with aqueductal stenosis or hydrocephalus in male and female fetuses. An important, very interesting diagnostic feature is that many times when we have obstructive hydrocephalus, we have stretching of the massa intermedia, the interthalamic adhesion, even detachment of it. But in L1 chem, in L1 syndrome, we have thickened massa intermedia and thickening of the quadrigeminal plate, the tectum. So look for these features also for the differential diagnosis. My king of the midbrain abnormalities is Joubert syndrome, one of the diseases that I dedicate the most. And you must say, Karina, you are quite crazy because we are speaking about ultrasound and you are showing us DTI. But this is the explanation for the molar tooth. And I think whenever we understand, we will remember it best. So here we see the normal control. We have the cortical tracts and the decussation, the crossing at the level of the superior cerebellar pedunculus and the corticospinal tracts in the normal one. What happens when it does not occur? So we don't have crossing of fibers, we have lack of tissue. So the interpeduncular fossa at the level of the corticospinal tracts is deeper, and the superior cerebellar peduncles do not cross. So we have this entrance together with the absence or the hypoplasia of the vermis, forming what we call the molar tooth sign. So this is a dysplasia of the brainstem, and the molar tooth dysplasias comprise Joubert syndrome and related disorders, as some other ciliopathies that concur with similar genes. This is an autosomal recessive inheritance disease with hypoplasia of the vermis and brainstem, especially midbrain dysgenesis. The related disorders will appear with abnormal kidneys, abnormal eyes, polydactyly, and other abnormalities. And there are many genes that are known. The mutation in the same gene may concur to different phenotypes, which is called pleiotropy. This is another paper that we have published on the normal aspect of the fourth ventricle. And this is a clue for many cases of Joubert syndrome that don't have the classic open fourth ventricle. We are used to scan the cerebellum at the level of the vermis, but I invite you to scan it in another additional plane at the level of the fourth ventricle. And fourth ventricle needs to be always larger laterally than longer anteroposteriorly. And whenever this proportion is inverted, as you see here, so you must suspect on a severe vermian dysgenesis. Joubert syndrome is the most important candidate, but many other abnormalities of the cerebellum and the vermis concur here. And we have published, we call it the fourth ventricle index, but the most important thing to understand is remembering always larger than longer. This is the classic open fourth ventricle, but in all cases that we don't see the opening, we need to check for the abnormality of the fourth ventricle. And of course, if we go sagittal, we see that the fourth ventricle is square shape because of the thickening of the superior cerebellar peduncles. We see no anatomical landmarks of the vermis, no primary fissure, no fastigium, and we see in many cases a very thick tectum, which is feature of the syndrome. Here we see the square shaped fourth ventricle compared to the normal in Joubert syndrome. As you see here also, the down-slanting cerebellar hemispheres, which I call normally, they are aligned, so I call them the seal, you'll see soon, and I call these the sea lion, so the down-slanting by the absence of the vermis, and this is a clue for Joubert syndrome. I'm just finishing. Butterfly sign is another very interesting mesencephalic finding, anterior cleft in the mesencephalon, which can be seen by MRI, but after we learn the pattern, we can come and see it also using ultrasound, again with the fusion of the diencephalic structures, so that the hypothalamus with the midbrain. Severino from Italia, she has published the different imaging patterns. We have no time to go into detail, but in type A, we will have the butterfly sign and the fusion of the hypothalamus to the midbrain structures, very hypoplastic midbrain, and in most cases, aqueductal stenosis with hydrocephalus. Also abnormal basal ganglia, and this is differential diagnosis with tubulinopathies. In type B, we see the connection between the massa intermedia to the midbrain. You remember that I asked you to check centrally here for the separation. So if you see communication and lack of cleavage, this is the encephalic mesencephalic type B. And look at the cerebellum, it looks like Dandy Walker. So sometimes the cerebellum may be very tricky. Go to the brainstem to make the differential diagnosis. This is also autosomal recessive with thalamic basal ganglia and brainstem abnormalities, and we need to differentiate from the tubulinopathies. Tubulinopathies may be very, very tricky, and this is my last topic. I'm showing you, this is late gestational age, 34 weeks, and please check for some of the features. This is the first feature. Whenever we speak about tubulinopathies, we need to check for brain asymmetry, not ventricular, but brain asymmetry, which is not like in megalencephaly, but milder. Here you see asymmetry between the ventricles, but not at the level of the atrium, at the level of the frontal horn. Squared shaped frontal horn, this is one of our first feature to suspect on the presence of tubulinopathy. You see also irregularity in the interhemispheric fissure. So very mild side differences. And also here at the level of the midbrain. And that's what I want to tell you, that is very, very tricky. So here, if you check on the side, use 3D multiplanar to be sure that you are facing a real, a true asymmetry between sides. There are very nice MRI papers that demonstrate this asymmetry. Look how finesse we go, but this is very important because sometimes these are the clues for the diagnosis. And the prognosis may be good, but sometimes may be bad. And this is very important for us to understand. The explanation comes from DTI, the thickness of the tracts and the crossing of the tracts and the difference between sides. So DTI, of course, explain, but won't be available to us. So remember our daily ultrasound to check for these details. I will leave you with my last slide. Some features for the differential diagnosis between dystroglycanopathies, L1 syndrome, and the tubulinopathies, remembering that those are the ones that normally appear earlier in pregnancy. Don't forget the rhombencephalosynapses as a differential diagnosis for early ventriculomegaly. Thank you very much, and I will be open for questions.
Thank you very much, Karina. There was a very the systematic midline lecture, including the many syndromes and pathogenic diseases. Thank you very much. Unfortunately, we have the discussion part, at the last part of this webinar. So I hope that you are staying with us. But if you have to leave earlier, we will miss you. But maybe I will let you know about the questions from the audience later on.
Thank you very much.
Thank you very much, Karina.
So our next speaker is Barbara Pertl from Austria. Barbara Pertl is professor of obstetrics and gynecology. She has a private clinic for prenatal diagnosis and risk pregnancies at the private hospital Graz-Rangnitz in Austria. Her medical education was at the University of Graz, Austria. She was research scholar at King's College, Guy's Hospital, London, and at Tufts University in Boston. She has been involved in several European research projects. Currently, she is the president of the Austrian Society of Medical Ultrasound. Dr. Pertl has published numerous scientific articles, including the introduction of a novel PCR technique for detection of prenatal chromosome abnormalities. She is an expert in prenatal diagnosis with a special interest in fetal neurosonography. The title of her presentation is Fossa Posterior Anomalies. Please, Barbara.
Thank you very much for this nice introduction. So, and also thank you very much for the invitation. The topic of my talk are fossa posterior anomalies. The fossa posterior contains the cerebellum, the pons, and the medulla oblongata. As part of the basic examination, the cerebellum can be assessed on transcerebellar plane. An algorithm was proposed for classification of the malformations of the fossa posterior based on this basic examination. So according to this algorithm, the anomalies of the fossa posterior can be divided into 3 groups. The first group encompasses anomalies with increased fluid-filled space of the posterior fossa. So the main anatomical landmark is the position of the tentorium, the docula, but also the rotation of the vermis and the anatomy and biometry of the cerebellum and the vermis. The second group comprises anomalies with decreased biometry of the cerebellum, so the reduction can be either focal or global. In case of global cerebellar reduction, you have to look at the brainstem, whether it's normal or abnormal. And the third group comprises anomalies with abnormal cerebellar anatomy. So considering this approach, it's very evident that the assessment of the cerebellum is of paramount importance for the diagnosis and also for the prognosis of posterior fossa anomalies. The basic exam is done in the transcerebral plane, so you can see very nicely the cerebellum with the two hemispheres and the vermis, the size and the shape of the fourth ventricle and the thalami. For further diagnostic workup, we have to do a so-called fetal neurosonogram, which includes additional planes. So the main plane, which is very important, is the mid-sagittal plane, which can be best accomplished by 3D ultrasound. Here you can see the corpus callosum, the sylvian aqueduct, and the vermis. And there's an additional plane, the coronal transcerebral plane. So in my talk, I would like to address group 2 of this algorithm, which means decreased cerebellar biometry with normal cerebellar anatomy. And I would like to present some cases from routine scans and also address the role of 3D ultrasound. So case 1 was an anomaly scan at 24 weeks of gestation. So the transcerebellar diameter was found to be below the 5th percentile. So on this graph, you can show this quite nicely, but if you look at this short video, the cerebellum looks completely normal. It even does look small. 2 weeks later on, the transcerebellar diameter was still on below the 5th percentile, and we also tried to assess the brainstem using 3D, and we also found the brainstem to be below the 5th percentile. So the MRI confirmed this diagnosis, so pontocerebellar hypoplasia was suspected. We did counseling in this regard, and we did an amniocentesis, mostly for whole exome sequencing, for identifying mutations in pontocerebellar hypoplasia-related genes. However, we always do also a chromosome microarray analysis, and in this case, we found a deletion on the short arm of chromosome 5, which is associated with Cri-du-Chat syndrome. So at the conclusion of this case, we can see there's quite a good agreement between the measurements obtained by 3D ultrasound and MRI. The next case, again, an anomaly scan. So we saw the transcerebral diameter within normal range, but a bit below the 5th percentile. But what was striking was a very low, quite low estimated fetal weight, which was on the 9th percentile. So 3 weeks later, the transcerebellar diameter started to drop, so the growth was slower through gestation. The baby was still small. So an MRI was performed and also a 3D ultrasound. So the transcerebral diameter was below the 5th percentile, and the vermis area was also small. This was also confirmed by 3D. So with 3D, we had also a very small vermis with all biometry data. So we counseled about, the patients were counseled about vermin hypoplasia. However, they didn't want to have any genetic testing because of the advanced gestational age. So the baby was born, and after 15 months, there was a really severe developmental delay, a secondary microcephaly, a cerebellar hypoplasia. On genetic testing, we had a normal karyotype and normal chromosome microarray analysis. However, with whole genome sequencing, we found a de novo cask mutation was found, which causes microcephaly with pontine and cerebellar hypoplasia. And the third case, we had again a transcerebellar diameter below the 5th percentile, but a normal pons, and in addition, a ventriculomegaly of 11 millimeters. So an MRI was performed. The Vermen area was still within normal range, but they found a smaller pons. And at 27 weeks, the Vermen area was quite well below the 5th percentile using 3D ultrasound. But what was striking was the appearance of the vermis. The vermis was, in this case, was very blurred, so you couldn't really see and distinguish the lobules and the fissures in comparison to a normal control at the same gestational age. So in this case, we did an amniocentesis and again found a combination of a deletion on chromosome 5 and a deletion of chromosome 21. So as a conclusion, there's a high incidence of genetic, also abnormal karyotype and pathological CNVs in cases with fossa-posterior anomalies. And this is especially true with Vermen hypoplasia and cerebellar hypoplasia. And the most common CNVs are the deletion on chromosome 5, 6 coterminal deletion, and X chromosome aberrations. So as a conclusion of these cases, it's very important to do a precise measurement of the transcerebellar diameter. A threshold of a transcerebellar diameter below the 5th percentile was suggested to identify fetuses at high risk for chromosome anomalies, major structural anomalies, and adverse outcome. 3D is of paramount importance to obtain a mid-sagittal view to assess the vermis. And fetal karyotyping and chromosome microarray analysis should always be offered. However, genetic testing alone does not solve the problem of counseling about neurodevelopmental outcome. So is there something else? So how can we assess the cerebellum and vermis using 3D? First of all, assessing the size. So the size of the cerebellum is measured on axial plane, and the measurement is done from outer edge to outer edge. The size of the vermis is measured by using 3D. So it's possible to measure the vermin height, which is the maximum superior-inferior diameter, which means this is the distance between the culmen and the uvula. And you can also measure the vermin surface area and their perimeter. There are a lot of different reference data for vermin height, but the cohort with the largest sample size was published by Cicchini, and I think these are very good data to measure the vermin height. And this was also a very interesting paper. They compared the measurements done with 2D ultrasound, 3D ultrasound, and MRI, and they found that the lowest you get the lowest results with MRI and the highest results with 3D. So the conclusion of this authors were that imaging technique-specific reference data should be used for the assessment of the vermis. But what about the integrity of the vermis? So the vermis is divided by fissures. This starts from 14 weeks of gestation. The fissures are best visualized in the vermis. The interval between the histologic appearance and the visualization on ultrasound and MRI is about 8 to 10 weeks. And it is said that in using ultrasound, you can visualize the primary fissure from 18 to 24 weeks and the secondary fissure in the 3rd trimester. But is this true? So on this graph, you can see a mid-sagittal, on this feature, a mid-sagittal view of the vermis. And you can see the different lobules. So this was published by the group of Daniela Breyer and Dovcak as a first author. And you can see the different lobules. There are 7 lobules because the declivifolium tuber almost all of the time appears a single structure. So instead of having 9, you can look for 7 lobules. Is this possible using 3D? Yes, it is possible. You can see in 3D the fissures appear as hypoechogenic structures, and the interlobal parts are hypoechoic. So you can see here the primary fissure, which is always very bright. So it's a very good landmark. Then here's the lobal, the DFD. Then here we have the prepyramidal fissure, the pyramus, the postpyramidal fissure, which is the secondary fissure, the uvula, and the nodulus. It is also possible to see at the time of anomaly scan. At the time of anomaly scan, you always, or most of the time, you get these pictures. So the primary fissure is very bright. Then here are the other four fissures, and you can also differentiate between the lobes. It's much better later on, so you can look at the differentiation and the integrity of the vermis later on better. So 23 and 24 weeks is a good gestational age. The best age to look at this is between 24 and 30 weeks of gestation, because then you can differentiate all 7 lobes and all the fissures as well. Later on it's quite difficult because of the bones of the skull, so it's sometimes very late, it's almost impossible to get a really good picture of the vermis. So we do an ongoing, we do a study. We analyzed 156 cases so far, and in very many cases it was possible to differentiate 5 to 6 lobules and 4 fissures. And given optimal image quality, it may be also possible to differentiate all 7 to 9 of all 9 lobules. So this work was done because of this paper, which was published by the Daniela Breyers and the Gregor Kasper's group with Dovchak as a first author. And they published a retrospective study. And after 22 weeks of gestation, they could discriminate 7 of the 9 lobules in the control samples. However, it was a retrospective study, so the image quality was sufficient for quantitative analysis only in 36% of it. And they also published a paper where they looked at this assessment for cystic posterior fossa malformations. They had a group 2 with Werman hypoplasia and a group 3 with Danny Walker malformation. And they showed that in these cases, it's only in 42% in the Werman hypoplasia cases, they could discriminate 6 to 7 lobules, and even less in the Danny Walker malformation group. So they suggested a cutout of 3 or fewer lobules as a possible indicator of rather worse outcome in cystic posterior fossa malformation. There was also another article published by XI. They did this with ultrasound, and they concluded that they could distinguish all 7 lobules from 25 weeks onwards. So what about our cases? So this is the case with the Cri du Chat. So we compared the pictures from the MRI with the pictures from 3D. And in this case, we could see quite a nice correlation, because with MRI, the specialists like Gregor Kasper, they told there are 7 lobules. And in our case, we found 5 lobules, which is normal with 3D. In the case with the CASC mutation, we had a very good agreement between the Werman area using 3D and MRI, and we hold also the same number of lobules. In the case of the Cri du Chat syndrome, we found less lobules by MRI as well as by 3D. So the conclusion of this is that 3D is particularly useful for assessing the size and the anatomy of the vermis. 3D assessment of the Werman lobules may be a useful additional tool to standard mid-sagittal measurements to assess the integrity of the vermis. Thank you for your attention.
Barbara, thank you very much for this excellent presentation. I think we'll have the question at the end of the session, and we'll have now a break of 10 minutes. So we see each other in 10 minutes.
everybody, if you have any questions or comments, please write down your questions and comments in Q&A box. Thank you very much. So see you later. Everybody, welcome back. So we will continue. The next speaker, next speaker is Vincenzo D'Addario, and he's from Italy, Bari, and he is the professor of the Department of OBGYN, University of Bari in Italy. And he has been involved in the research and education of the ultrasonographic diagnosis of the fetal central nervous system for many years. And I actually know that I learned from him a lot, for many years. So Vincenzo, please start your lecture.
Thank you, Ritiko, for your kind words, for your presentation. Thank you, Ebert, also for your invitation to participate to this webinar, which gave me the possibility to listen to the high-level presentation by the previous speaker. My topic is ventriculomegaly. Ventriculomegaly is a condition which relates to a spectrum of pathologies characterized by dilatation of the lateral ventricle more than 10 millimeters. It is important to measure correctly the atrial width following the rules suggested by International Society of Ultrasound and Gynecology, which consider to measure the atrial width at the level of the glomus of the thyroid plexus, positioning the calipers on the inner border of the lateral and medial wall of the ventricle. According to this measurement, ventriculomegaly may be defined as borderline or severe, borderline when it is between 10 and 15 millimeters, severe more than 15 millimeters. It is useful also to divide the borderline group in two subgroups: the mild form with the atrial width between 10 and 12 millimeters, and the moderate form between 12 and 15 millimeters. This differentiation is useful for counseling, as we will see later on. Screening for ventriculomegaly is relatively easy. High sensitivity has been reported, but this high sensitivity refers to severe ventriculomegaly and only severe ventriculomegaly, and also it is diagnosed late in the third trimester. Before 24 weeks, the sensitivity drops to 35%, reflecting the natural history of the disease. And when you screen for ventriculomegaly, it must be taken in mind that the ventricle proximal to transmitter, even using high-level machines, is frequently obscured by the reverberation aspect. By tilting the probe is supposed to recognize also the proximal ventricle, in this case of unilateral ventricle. Ventriculomegaly is easy diagnosis, but is not a final diagnosis. It is a common finding common to different conditions, ranging from normal variants to severe diseases with poor neurodevelopment outcome. When ventriculomegaly is diagnosed, we have to rule out for associated anomalies, for associated chromosomal and genetic diseases, and for infections. Associated anomalies, both cranial and extracranial, are more frequent in cases of severe ventriculomegaly. Percentage ranging from 58 to 65%, but it may be present also in borderline ventriculomegaly, with percentage ranging from 30 to 51%. For this reason, careful sonographic examination of the fetal ventriculomegaly must be performed, and severe brain anomalies may be recognized by a detailed neurosonogram. The neurosonogram is obtained by using not only the axial view with the transventricular, transthalamic, and transcerebellar planes we have used routinely in the second trimester, but extending the evaluation also to sagittal view, the mid-sagittal and parasagittal one, and coronal views from the anterior to posterior aspect of the brain. And this allows to visualize all the ventricles, the corpus callosum, cavern septicity, the cavern complex, the thalami, the terminal matrix region, the posterior fourth, and so on. The evaluation of the brain in the three spatial planes can be easily obtained by dimensional ultrasound. After obtaining a volume of the fetal brain, the multiplanar view can be used, and the three planes are rotated in order to obtain the mid-sagittal one, showing the mid-sagittal pictures, in this case, the corpus callosum, cavern septicity, the cavern verde, and the cerebellar verde. Tomographic ultrasound imaging can be also used to obtain multiple parallel sections in different planes. In this case, we can see a mild ventriculometry and unilateral ventriculometry. The CNS anomalies which can be associated with ventriculometry are several. The most common are aqueductal stenosis, agenesis of corpus callosum, belly wall malformation, chiari tumor formation, occupying space lesions, cortical malformation, heterotopias, hemorrhage, and many, many others. For this reason, an accurate evaluation of the brain must be performed for when the ventriculometry mild or severe. Aqueductal stenosis is the cause of the some of the so-called isolated ventriculometry. Aqueductal stenosis may be primitive with multifactorial pathogenesis. In this group, this link of the hypercephalus is included. May be secondary to post-infective gliosis in cases of toxoplasma body infection, secondary to hemorrhage or to external compression by tumor system vascular. The sonographic patterns of ventriculomegaly these are the characteristics. We have an isolated dilatation of the lateral ventricle with mild or even after dilatation of the ventricle. The normal the fortiform ventricle and the cerebellar verde, the corpus callosum at normal. This is a multiplanar visualization of the severe ventriculomegaly due to occipital stenosis. The dilatation of the third ventricle is easily recognized as the corpus callosum is normal. However, the dilatation of the ventricle may be only maybe very minor in these cases, or can be suspected when midline cyst is visualized in cases of ventriculometry, which is referred to dilatation of the suprapineal recess, which is a recess in the posterior aspect of the severe obstructive ventriculomegaly produces also an increased circumference, parenchymal thinning, decrease of absence of the peripheral spaces, and penetration of the midline. Aqueductal stenosis may be also secondary to an intraventricular hemorrhage. Signs of hemorrhage are easily recognized. We have hyperechogenic walls of the lateral ventricle, echogenic material in the lateral ventricle referred to blood clots, and we can also have brain damage in cases of cerebral hemorrhage. Severe ventriculomegaly with fenestration may be also the consequence of the encephalostenosis, a condition where the aqueduct is formed because of the fusion of the two encephalon. In this case, typical cerebellar butterfly sign due to the dilatation of the brain. Another condition which can cause ventriculomegaly is corpus callosinogenesis. I think that the next speaker will speak about this condition, so I will briefly remind you the sonographic signs, which are several. Colpocephaly is typical. Dilatation of the uterine space, which is another typical sign. Absence of the corpus callosum in the sagittal view. Bullsed appearance of the frontal horns in the coronal view. Dilatation of aperture. My ventriculomegaly may be also associated to a very rare condition, the thick corpus callosum, which can cause, which is been described in the postnatal period, associated with psychotic. Another condition which can cause ventriculomegaly is chiari malformation, a condition which starts from spinal defect and through the leakage of spinal fluid, the compression of the ventricular cavity, lack of distension of the lumbar cavity, development of a small posterior cortex, and the obstruction of the spinal space produces in 80% of the cases of malformation produces ventriculomegaly. In this case, we have the very well-known associated signs of chiari malformation, which are the dysmorphic cerebellum with the banana sign and the obliteration of the coccygeal. In the Walker malformation, ventriculomegaly is consequence of severe cranial hypoplasia, cystic dilatation of the fourth ventricle, tentorial compression, obstruction of the supratemporal artery spaces, and finally the ventriculomegaly. The sonographic signs are the fetal ventricular dilatation, distal posterior fossa, widely communicating from the fourth ventricle. In the sagittal view, we recognize an apoplastic as atrophic rotator cerebellar fragments and an elevated. Joubert syndrome is already said by the previous speaker, may be a cause of severe ventriculomegaly, such as but the list of conditions, cerebral anomalies, complicating ventriculomegaly are seven. We can have also neuronal migration, proliferation disorder, in this case, microencephaly, and these are periventricular. Also, tumors may cause dilatation by compression of the artery. Ventriculomegaly, mild ventriculomegaly may be associated with interhemispheric cysts like this cases of carbon-based intercostal cysts or this case of and finally, this unusual case of choroid plexus bifidum, which causes mild ventriculomegaly. In case of ventriculomegaly, the request of the MRI has been suggested. A meta-analysis of some years ago demonstrated that the MRI added more clinical information in 10% cases of the ventriculomegaly diagnosis. And more recent paper published called Meridian for the group, which reported an accuracy of ventricle of MRI superior to the ultrasound, 98% MRI in comparison go to consider, if we consider the anomalies which were reported as additional finding by MRI, in 55% of the cases, there are agenic opportunities for the cortex callosum. In 6% absent of cortex callosum sexual dysplasia, which are conditions which can be easily recognized by us. In fact, if we consider the clinically relevant information on MRI, reported in 20 studies collected from the literature by Dario Palatini, it appears evident that this percentage was only 5% in cases when the examination was done by a fetal medicine doctor or physician in comparison to 10% reported by ultrasound performed by radiologists. That means that also using the translational approach, we have much better results in translation. That means that an accurate neurosonogram is the basis to decide whether to ask for MRI or not. Anyway, the condition, the brain conditions where MRI is superior to ultrasound are ingression disorders, parenchymal damage, infarction, terminal matrix. Another very important topic is the association of ventriculomegaly with the chromosomal. This association is present in 2 to 17% of the cases, depending on atrial reaches and the presence of other structural in the case of tracking 18 in fetus presence of ventriculomegaly, severe ventriculomegaly, interventricular septal and palsy. In cases of mild ventriculomegaly, the association or isolated mild ventriculomegaly association with the chromosomal is lower. 3 meta-analyses reported the percentage of association ranging from 2.8% to 5%. The variation of results may depend on the prevalence of pregnancy in study population, which in turn depends on previous life screening program. Now, it is very common to use fetal DNA in the screening for sonography, and the residual risk of pregnancy in the context of ventriculomegaly and low risk fetal DNA is still unknown, but counseling should be given on the fear risk of other chromosomal abnormalities not regulated by fetal DNA, especially if compared to the diagnostic possibility of a microarray study. Microarray studies can find copy number variants in fetus with pregnancy ventriculomegaly non-serotypes in 5-10% of the cases, which percentage higher in non-isolated in comparison to isolated cases. So, diagnostic samples, which is CGH should be offered when ventriculomegaly is detected. And even in cases of normal results, patients should be informed that tests cannot detect all genetic causes of fetal ventriculomegaly. Because there are some causes of ventriculomegaly related to the gene system. So, in cases of ventriculomegaly, genetic counseling is recommended. Another topic is the association of ventriculomegaly with fetal infection, and mainly toxoplasmosis characterized by the presence of small periventricular calcification or cytomegalovirus infection, where we can find sometimes multiple tuberculosis. As regards the outcome, the long-term outcome is mostly influenced by the basic disease and the accompanying analysis, the degree of ventricular dilatation, and the progression of ventricular dilatation during treatment. These are the outcome different causes of ventriculomegaly as reported by the pediatric point of view after delivery. In after stenosis, we can have a good outcome when it is isolated in more than 80% of the cases. In carry tumor formation, the main problem is the motor and stereo damage due to stenolysis, but IQ is reported to be 75 to 85% in the good in 75 to 85%. In daily water malformation, the prognosis is variable, mainly depending on the associated anomaly that is usually poor. In callous genesis, the prognosis is variable, mainly depending on the associated anomalies, and is reported good in 70% of the isolated cases. In cases of occupying space deletion, the prognosis is depending on the size of lesion, is tumor damage, and in cases of focal malformation, the prognosis is poor. These are the results of a meta-analysis of the outcome of people with isolated severe bilateral ventriculomegaly. This meta-analysis reports a percentage of 12%. Normal development is providing 42%, which is much less than the percentage reported by postmortem literature, and it depends on the natural history, because in the postnatal literature, only the fetuses which arrive to normal delivery and which are related to ultrasounds have this good outcome in comparison to the poor outcome reported in the prenatal series. Severe disability is reported in 40% of the cases. Different is the condition in cases of isolated mild ventricular. Neurodevelopment delays in usage locomotor activity, eye and hand coordination, autism, epilepsy, hearing and speech capacity, and learning performance is reported in 3 different meta-analyses in percentage ranging from 7.9 to 10.9%. But if you consider the cases of mild, moderate ventriculomegaly, you can count that this is associated with the association of poor outcome neurodevelopment delay is only 5% of mild ventriculomegaly and 17% in moderate ventriculomegaly. And in mild ventriculomegaly, a recent review reported no significant difference between a group of fetus diagnosed with isolated mild ventriculomegaly and the reference population, which percentage of the overall neurodevelopment delay of 5.7% compared to 2.2%. Another important point in the evaluation in the evaluating the outcome of the fetus with ventriculomegaly is the progression, stability, and resolution of the mild ventriculomegaly. And of course, the outcome is worse in cases in the 50% of the cases when ventriculomegaly progresses during pregnancy. These cases, in fact, we have a normal outcome of 44% in comparison to the 4% of cases where the ventriculomegaly decreases and 80% that when ventriculomegaly remains stable. Counseling is easy when the postnatal outcome is surely severe. It's easy to say that the outcome is poor in cases of severe ventriculomegaly penetration of the nasal. But the counseling is uncertain when the prognosis is known to be variable as in cases of isolated corpus callosum. This justifies the 8% of the mutant pregnancy, even in cases which could potentially have recovered. So the message to take home is that fetal ventriculomegaly is a native recognized sonographic pattern. We have to search for associated anomalies, we have to perform karyotype in the CGH array, particularly cases not isolated. We have to search for fetal infections. We have to counsel the parents according to the presence of a fetal anomaly, severity and progression of the fetal development. And finally, we have to inform the parents on the possibility that some anomalies can be diagnosed. Thank you for your attention.
Vincenzo, thank you very much for this comprehensive presentation on ventriculomegaly. We move on to the next speaker, Sonila Pasha from Frankfurt, Germany. Dr. Pasha had her medical education at the Medical University of Tirana, Albania. She was teacher at the Albanian University for several years. In 2012, she graduated in Germany at the University of Mainz. Her doctoral thesis was about 3D ultrasound, normal and abnormal development of the fetal corpus callosum. Since that time, her main interest is 3D ultrasound of the fetal brain. She also published growth charts of the corpus callosum with 3D ultrasound. Since 2017 until now, she is working with me at the Center for Ultrasound and Prenatal Medicine in Frankfurt, Germany. She published several scientific papers on the fetal brain, and she got several awards. She is editor of the international journal Ultrasound International Open. I can say she is a true expert in 3D ultrasound with special interest in fetal neurosonography. She is now going to talk on the corpus callosum and cavum septi pellucidi, normal and abnormal findings. Sonila, please.
Thank you very much for the introduction. The aim of this presentation is to outline the practical approach in the demonstration of the corpus callosum with 3D ultrasound, as well as to give some clues in the pathological cases. Corpus callosum is the major connective pathways of the human brain. It consists of more than 190,000 myelinated fibers located deep in the longitudinal fissure connecting the left and the right hemispheres of the brain. Before I get into the prenatal diagnosis, let me talk a little about the anatomy and embryology of the structure. Corpus callosum segmentation in a clockwise order, brain looking from the left, are rostrum, genu, body, and the splenium. Depending on whether the callosal fibers are connecting the associative areas, they are poorly and small, myelinated, and if they are connecting primary motor areas, they are heavily and large myelinated. Why is this important? We have here many times that the corpus callosum is a structure, but within the structure we have different fibers that have different function. That's why it's important that this structure has to be showed correctly. And in this point is the 3D ultrasound the clue of the exact demonstration of the corpus callosum. In the field of the embryology, we have also some conflicting data. If I am consulting a patient, they frequently ask, is that the corpus callosum the only pathway that connects the left and the right hemispheres of the brain? Of course not. We have other connections, and these are commissure anterior, commissura fornix, is corpus callosum, and cavum septi pellucidi. But why are this structure important? Here we have a schematic presentation of the embryologic development of this midline connection fibers, and we see that the commissure anterior is development at 7 to 9 embryologic weeks, and commissura fornix at 9 to 10, corpus callosum at 11, and capum septi pellucidae at 12 till 14 embryonic weeks. This is important for the diagnostic of pathological cases, and I will come back to this slide at the part of the pathological ones. So corpus callosum will be interesting in many scientific disciplines. We have conflicting data in embryology about the way the corpus callosum developed. They are saying in the later studies that the corpus callosum developed in the midline, in the hippocampus area, and then developed bidirectionally. But in the studies we have seen before, they are saying that the corpus callosum is developed from front to rear, with the rostrum developing the last. And these clues are important always in the diagnosis of pathologies. But let's move on to the prenatal diagnosis. In the prenatal diagnosis, we also have conflicting data about the echogenicity of this structure. At the beginning, when the studies were performed just with 2D ultrasound, corpus callosum was explained as a hypoechoic to anechoic structure. But lastly, as you see in this publication, with the mean of 3D ultrasound, corpus callosum is demonstrated as a hyperechoic structure. I would like to explain why are these studies and how we can use the 3D ultrasound without changing the echogenicity of the structure. So we can use 3D ultrasound because we have the 3 perpendicular planes, and this help us to be oriented exactly in the mid-sagittal plane. And you can see here we have 2 pictures. The first one is with 2D ultrasound, and the second is with 3D ultrasound. Both of them shows the corpus callosum as a hypoechoic structure, but in the 2D ultrasound, I am not in the midline, because if you see here, we are a little bit oblique in a plane, and in the 3D ultrasound, we have a very good picture of the brain tissue at the surface. So that's why it's important to use 3D ultrasound. But what is the profit of this method? First of all, if we see here in 2D ultrasound, we are able to take oblique planes, and in 3D ultrasound we are able to take a volume. That means that within a volume we are able to have parallel planes as well as the oblique ones. But what is the difference? Do they use other methods? No, we use the same acoustic window, and for the study of the fetal brain, we use the sutures and the fontanelles. And for the corpus callosum, the most recommended one is the metopic suture, sagittal suture, or the anterior fontanelle. So, as I say to you, we use 3D ultrasound because we are able to have the 3 orthogonal planes. And with that, we can see that here we have in a vertical position and here also in a vertical position. And that reassures us that we are in the midline of the fetal brain. Furthermore, we can use the surface render mode in a way to determine a better and to have a better visualization of the midline structures of the fetal brain. So as you see here, I am at the midline structure of the fetal brain. And if I would like to know for where I take this volume, I can use the surface rendered mode with the maximum mode, and I can see that I am exactly in the midline and I am using the anterior fontanelle to have this better image of the corpus callosum. But can I use just the parasagittal or the sagittal planes for the demonstration of the corpus callosum? No, I can also use the coronal planes from the anterior fontanelle. But in this way, I need a big angle, and that means that if the fetal is moving, I cannot have a better quality of the corpus callosum. That's why if we are taking the picture from the parasagittal or sagittal planes, we have a better resolution and we need a very small angle. And if we are from this plane, coronal one, we need a very big angle, which sometimes it's impossible to have during our examinations. If the baby is in the breech presentation, what are our possibilities to arrive at the anterior fontanelle? So first of all, I will recommend that you can try to move the patient at the left or at the right, but if it's not functioning, then I will recommend the sitting position as in this figure. So I have a very good experience in the demonstration of the corpus callosum with this kind of position in the breech presentation. What I will not recommend, it's the demonstration of the corpus callosum from the axial planes. We use axial planes for the biometrical measurements that we perform routinely, and many professionists are recommending this plane as a useful one for the demonstration of the corpus callosum. And even if I take a plane, a volume from the axial plane, and I use different displace mode of the 3D ultrasound, it is also not possible to demonstrate the corpus callosum. As I say to you, I have used this image to have the corpus callosum, and you see the A and the B plane. You can have some information from the patient, but the C1, it's a reconstructed plane. That means that you have artifacts, and you see here that the corpus callosum is a hyperechoic structure, and you cannot detect where is the rostrum, where is the genu, truncus, and the splenium, because all of them are connected and demonstrated as one structure. So you can see also here, if I am using the tomographic display mode, I also can see some tissue of the corpus callosum, but I cannot say whether it's a partial agenesis, whether there is an artifact, and that's why I will not recommend this plane for the demonstration of the corpus callosum. Also, if I am using the OmniView, here we have the same problem. So we see that the corpus callosum is demonstrated as a hyperechoic area. And a hyperechoic area is a pathology of the corpus callosum, which is called lipoma of the corpus callosum. And this cannot be so often as we see. So please do not use in your clinical daily practice the axial plane for the demonstration of the corpus callosum. How I do it? So for the demonstration and 3D use of the corpus callosum, it's important to have a sagittal or a parasagittal plane. That means that if I am in the middle, I need some of these planes. And if I am there, I need to press just the 3D button and then to take the volume. During the volume acquisition, it's important to observe on the monitor that you have all parts of the corpus callosum included in the volume. And then you can manipulate the volume. How is possible? So what you have to use are this reference dot in all three planes. I personally use the coronal plane for the orientation and finding of the corpus callosum. So first of all, I use the interhemispheric fissure to place the dot. And then I rotate in the coronal plane the z-axis to have a vertical orientation of the coronal plane, as you see here. And after that, I correct also the axial plane of the fetal brain, also with the z-axis. And after that, I can have the exact median plane of the fetal brain. And I cannot observe just only the corpus callosum, but also the other structures that are present, like fornix in this picture, or the vermis of the cerebellum, and so on. So I would like to emphasize that the 3D ultrasound is a very good tool in the demonstration of the corpus callosum. I will recommend to use the anterior fontanelle because you have a big acoustic window that you can use throughout gestation for the demonstration of the corpus callosum. The second one, acquire a volume for a parasagittal, coronal, or a median plane. Then use the multiplanar display mode for the anatomical orientation within the fetal head. And please do not use the axial planes for the demonstration of the corpus callosum, because this will lead to artifacts and not a correct demonstration of the structure. So during our studies, we performed measurements of the corpus callosum by means of 3D ultrasound. And as I say to the anatomical part, it's important for us to observe whether all parts of the corpus callosum are there and all three lengths, because the corpus callosum is a curved shaped structure. That's why we are able to confirm the normal development just by means of three different measurements. And we perform the inner, the outer, and the curved measurements. As you see here, we also perform the measurements of the height of the different anatomical parts of the corpus callosum. As to what we observed that the corpus callosum have a different development. So we have some changes in the vertical orientation of the rostrum. As you see here, with 17 gestational week, we have a very semi-vertical orientation, but at the end, we can see here that the rostrum has a semi-horizontal orientation. We also see the development of the genome, which is at the beginning not a prominent part, but as you see here at the end of the pregnancy, it will become more thicker, and it will also have some flexion. So what about the definition of corpus callosum pathologies? Here we have also many conflicting data in the literature, how we can define the different corpus callosum pathologies. We have no problems with the corpus callosum agenesis, because this is a clear identification. And if we do not see any corpus callosum, this is the complete agenesis of the corpus callosum. But in some cases in the literature, partial agenesis of the corpus callosum, hypoplasia or hyperplasia, are included in corpus callosum dysplasia. And this makes our cases difficult to interact between different professionals, and also to follow up the pathological cases. That's why, according to our growth charts, we decided to have a very clear definition. The partial agenesis of the corpus callosum was diagnosed when one of the parts of the corpus callosum is missing and all three lengths are less than the 5th percentile. And hypo and hyperplasia were diagnosed where all three lengths were within the normal range, but the most important one, all parts of the corpus callosum has to be present, and one or two parts of it has to be more or less than the our normal range. What is important in the diagnosis of corpus callosum agenesis? If we are using the axial planes in our routine daily work, it's important not to misinterpret the cavum, the fornix, as the cavum septipellucidum. So you have always to be sure that you have a hyperechoic structure in the anterior part of the fetal brain. And if you are seeing these three parallel lines, then you are a little bit below in the brain. And that means that you can miss the diagnosis of absence of the cavum septipellucidum. Why is important? At the beginning, I say to you that the corpus callosum comes before the cavum septipellucidum in the embryological development. What means that? This means that if we have an agenesis of the corpus callosum, the cavum septipellucidum will also be absent because it comes as a later event in the embryology. But if we have an absent cavum septipellucidum, we can have a normal corpus callosum, we can have a pathology of the corpus callosum, but we can have also an agenesis of the corpus callosum. So it is very important to diagnose also if the cavum septipellucidum is present or missing. What we use in the prenatal diagnosis of complete agenesis, there are some indirect signs that both, I hope that all of you are aware of it. And here you can see some cases when we can diagnose the teardrop-shaped ventricles. The absence of the cavum septipellucidum is in all cases when we have an agenesis of the corpus callosum. We have increased interhemispheric fissure, upward displacement of the third ventricle, and a radial array of the median sulci, but this is diagnosed just in the third trimester. So it's another point that we always have problem in consulting our patient. It's the outcome of this fetuses, and these are always depending on the associative malformation. And even if we have an isolated case, we can not guarantee the couple that the baby will be normal, because maybe they have some migration disorder that are not possible to diagnose in the prenatal period and can come at a later stage. Partial agenesis of the corpus callosum is diagnosed when one of the parts of the corpus callosum is missing, and you can see here it can miss the posterior part, it can miss the anterior part, it can be missed also the body or the truncus of the corpus callosum, and this different malformation, partial agenesis, can also support the thesis of bidirectional development of the corpus callosum embryologically. What I find interest is that in many cases when we have a partial corpus callosum agenesis, we have accumulation of some cystic structure. So if you see some cystic structure in the midline, please go and control whether the corpus callosum looks normal. In cases of hypoplasia, we have an event that can result at the late destruction of the callosal action. And we can have metabolic disorder, infection, or obstruction. And according to our cases, these are the cases that have also the worst prognosis, because there is something genetically or metabolic that doesn't allow the brain axon to develop normally. In cases of hyperplasia of the corpus callosum, it can be diagnosed in schizophrenia, neurofibromatosis, macrocapillary malformation syndrome, and Cohen syndrome. And if you see also publication in the literature, you can see that just one of these babies is developing normally. It's also, this kind of malformation can also be found in every part of the corpus callosum, but sometimes can be also a transitory finding. So please, if you see an hyperplasia of the corpus callosum, go and control the development of the structure. And lastly, as I say to you, if you see in the midline structure an hyperechoic structure, this is a lipoma of the corpus callosum. So be aware of this malformation because it will help you in the diagnostic of this kind of malformation. And what I will recommend, if you make a diagnosis of corpus callosum pathologies, I will recommend to talk with the couple and to explain them that many of this malformation have association with chromosomal anomalies. So I will recommend, first of all, to perform a fetal karyotyping, and if it's possible for the couple, depending on the culture and on the insurance support of different countries, one can also perform a chromosomal microarray and an exome sequencing. If it's positive, then the couple can be consulted according to the etiology. But if it's negative, you also have to say to the couple that in 20% of cases, there is a residual risk for disability. So in conclusion, I can say that the 3D ultrasound is an excellent tool in the demonstration of the corpus callosum. The volume should be acquired in sagittal or parasagittal plane. Corpus callosum can be demonstrated as a hypoechoic or anechoic structure throughout the entire gestation. A diagnosis of corpus callosum agenesis can be performed as early as 20 weeks of gestation. Knowledge of the normal development can help us to make a clear diagnosis of corpus callosum pathologies. In cases of diagnosis of pathologies of this structure, you have to check up for further diagnostic like genetic consultation, because this structure, it's also have some problematic in the development and also in the outcome of the babies. And with that, I would like to thank you for your attention.
So thank you very much for this comprising presentation. Now I give the micro to Ritsuko.
Okay, so the next speaker is Professor Eberhard Meltz. Your main lecture of this the seminar, and Professor Meltz is a clinical pioneer of 3D ultrasound in OBGYN, and actually he's a father of the 3D ultrasound, and he has embarked on the development and use of the 3D three-dimensional sonography, and so please, Father.
Ritsuko, thank you very much for the kind introduction. Well, the term syndrome, it's a set of medical signs and symptoms that are correlated with each other. The term syndrome originates from a Greek word that combines the root to run with the prefix together. So we always have to look for several abnormalities when we are looking for the fetal anatomy. Well, if you look at the data from the EuroCAD registries, where they presented congenital cerebral anomaly cases, you can see that in approximately 26% of cases you will find a chromosomal defect, a genetic syndrome, or a teratogenic syndrome. So you have in 26% of the cases, you have a syndrome. So that's why it's very important to look for several details in the fetal anatomy. What can we see in ultrasound? Which brain abnormalities we can find? First of all is microcephaly. We can see a dilatation of brain ventricles, as we have seen from Vincenzo da Dario. Abnormal shape of brain ventricles. We can find absent septum pellucidum or absent cavum septi pellucidi. We can also find choroid plexus cyst, abnormal cerebellum, as we have seen from Barbara. And we can also have abnormal migration or gyration. And we can also find encephalocele and agenesis of corpus callosum. The most severe malformation is microcephaly, of course. Here with 3D ultrasound, with a surface demonstration, you can see the normal shape of the fetal head. And here in microcephaly, you see immediately there is a part, the upper part of the skull is missing, and also the brain is too small. And here you can see there are several syndromic causes of microcephaly. You can find chromosome abnormalities like trisomy 21, 13, 18. You can also find gene deletion syndromes like Wolff-Hirschhorn syndrome, Williams or Miller-Dieker syndrome. And you can also find other syndromes with multiple anomalies like Cornelia de Lange syndrome, Smith-Lemli-Opitz syndrome, or also holoprosencephaly. Of course, we are using biometry to make sure that this is a true microcephaly and not a pseudo microcephaly. The dilatation of the brain ventricles. There are several syndromes that are associated with ventromegaly. It's, for example, the Uppert syndrome, the Fragile X syndrome, the Robert syndrome, Smith-Lemli-Opitz syndrome, and the Gruchy syndrome. That is a deletion syndrome, ATP minus. Here, for example, you can see that was a dilatation of the brain ventricles. And if you look in detail, you can find there is a subtle cleft in the skin here. And here you see the baby after delivery. And that was a de Gruchy syndrome. It was a growth retardation, and you will find craniofacial and brain malformations. Abnormal shape of the brain ventricles. The most severe you can find in holoprosencephaly, whereas the allobar holoprosencephaly, you have a complete failure of the forebrain to divide into the two hemispheres. You can find holoprosencephaly in Patau syndrome, Edwards syndrome, Smith-Lemli-Opitz syndrome, and Hartfield syndrome. Here you can see the variants in holoprosencephaly. This is the normal brain, the allobar holoprosencephaly, the semilobar, the lobar, and the middle variant. With a view from a bird's eye, you can see there is, in this case here, it's an allobar holoprosencephaly because you can only find one semicircular ventricle in the front part of the fetal brain. Another sign that was already mentioned by Sunila Pashai, this is the so-called teardrop sign, where you can see the ventricles have the shape of a teardrop. And also you can see here where the distance in the front part is larger here to the midline than in the posterior part. And this is a typical sign for the Aicardi syndrome with agenesis of corpus callosum, as you can see here, and chorioretinitis abnormality. Another sign is the choroid plexus cyst. You can find choroid plexus cyst as a unilateral or a bilateral finding. Here you can see on both sides, you can see big cysts, more than 10 millimeters on both sides. And if you are doing a karyotyping, you have the chance to find a chromosomal defect. In that case here, it was trisomy 18. Here, if you are looking whether it makes a difference, whether the choroid plexus cysts are unilateral or bilateral, you can see there is no big difference. In both cases, you will find a chromosome abnormality in approximately 3.6%. Here you can see unilateral 3.3% and bilateral 3.9%. What you can find in most cases is trisomy 18, but you can also find in some cases trisomy 21. Absent septum pellucidum. If you are looking from the frontobiparietal diameter here, and you are once again, you are looking from a bird's eye view, you can see in the front side that the septum pellucidi is missing completely. You see there is a direct connection between the two frontal horns here. And what you can also find in such a case, this is the hypoplastic optic nerve. Here you can see on the right side, while on the left side it's a normal optic nerve here. There's the septo-optic dysplasia, is also called de Morsier syndrome, and this is a triad of optic nerve hypoplasia, hypothalamic hypophyseal dysfunction, and midline abnormality. Well, the so-called septo-optic dysplasia plus syndrome. This is a combination of the septo-optic dysplasia and a schizencephaly. Schizencephaly is also a rare birth defect that is characterized by abnormal cleft in the brain. This cleft can occur bilaterally or unilaterally, and you can see here this lateral cleft here on the left side here. This is unilateral here. In those cases, there is a genetic association with the EMX2 gene, and in also some patients mutations in the COL4A1 gene. So in all these cases, of course, we need a genetic examination. Abnormal cerebellum, we also have heard before. It's the most often demonstration is in the Dandy-Walker malformation, where you have an absent or small vermis. You can see this indentation here in the middle, where the vermis is missing, and you only can see the lateral parts of the cerebellum with an enlarged posterior fossa. Abnormal migration and gyration. This is the most complicated part to detect with ultrasound. Well, you see the development of the cerebral cortex is rather complicated, and you will find the development in 3 different stages. One is the cell proliferation, the second is cell migration until 20 weeks of gestation, and the last is the cortical organization that takes place after 22 weeks of gestation. If there is any problem in this development, you will have disorders with in these abnormalities of gyral formation and also of cortical organization. With ultrasound here, with 3D ultrasound, you can see we can precisely demonstrate the process of gyration. Here you can see with 21 weeks of gestation, the brain bundle is very smooth. Also with 27 weeks of gestation, it's still smooth. However, after 34 weeks, you can find this folding of the brain here, this gyri, and with 37 weeks of gestation, you see all these different gyries. That is a normal development. we can also use the coronal view. There we can differentiate, like in the anatomy, between the gray matter and the white matter. And in that case, we are also able, we can demonstrate also abnormal neuronal migration. In this case here, you can see there is a periventricular nodular heterotopia with, you see these white dots here in the surrounding of the ventricles. And also here, if you look, if you push the brain through this window, you can see here in the surface demonstration, you can see this hyperechoic structures surrounding the ventricles on both sides here. Another abnormal neural migration is lissencephaly. This is a gyra or pachygyra complex. We can differentiate between two types of lissencephaly. One is lissencephaly type 1. It's the isolated lissencephaly or the Miller-Dieker syndrome. This is a deletion syndrome. Or type 2 lissencephaly, the Walker-Warburg syndrome, the Fukuyama muscle dystrophy syndrome, or the muscle eye brain disease. Well, it's important to know that genetically most of the lissencephaly cases are caused by mutations of two genes. It's the LIS1 and the DCX or XLIS gene. So it's very important also to have here genetic examinations. Well, the Miller-Dieker syndrome type 1, lissencephaly type 1, this is characterized by an abnormal brain development. You have an abnormally smooth brain with only a few folds and grooves. Here you see the normal brain here, and this is the lissencephaly brain, where you have a very smooth brain mandle here. The same you can demonstrate with 3D ultrasound. It's not necessary to have an MRI for this diagnosis. You just have to use the surface mode in 3D ultrasound, and you can demonstrate the same malformation as like with MRI. Here you see the normal brain with a normal gyration, and here you see another brain with 38 weeks of gestation where there are absolutely no gyri to see. This is a very smooth brain mandle in case of a lissencephaly type 1. You see, it's a very rare condition, and in that case, it's not easy to detect this malformation. This is another case of Miller-Dieker syndrome with lissencephaly type 1, where you can see again pachygyria. You see here, this is a smooth brain mandle. You can find microcephaly. You can find hyperplasia of corpus callosum. You see the corpus callosum here is very thin. You can find the dilatation of brain ventricles, as you can see here, prominent forehead, mid-face hypoplasia, as you can see here. You can see only one nostril, and also you can find an anti-mongoloid Iceland. Furthermore, you can find low-set ears and cataract, as you can see here, and also here you see the low-set ears that are practically next to the shoulder here. Lissencephaly type 2 is the Walker-Warburg syndrome. This is a syndrome with an obstructive hydrocephalus and severe other malformations of the brain. Walker-Warburg syndrome has been linked to at least 14 different genes that are responsible for making the proteins for the glycosylation process. In that case, you can also find hydrocephalus, you can find the posterior encephalocele, pachygyria, or also called cobblestone cortex here. Eye malformations you have in 100% of the cases. You can find microphthalmia or cataract, as you can see here. You can find muscle dystrophy. This is not possible to detect with a 3D ultrasound, only if you have a very low movement of the fetus, but you can also detect malformations of the cerebellum that will occur also in 50%. Here you see another severe Walker-Warburg syndrome, where you have not only severe hydrocephalus, but you also a severe fenestration here between the two hemispheres here and the two ventricles here. And if you're looking from a side view, you can see exactly the hole that is in the falx cerebri here. And as you can also see here, you have the detachment of the retina on both sides here. Agenesis of corpus callosum. This is the Aicardi syndrome with also a very low incidence with 1 to 100,000 live births. Here you can see the agenesis of corpus callosum. You can find cleft lip, cleft palate, and also in some cases you can find rib or spine abnormalities, like you can see here, in this case with the hemivertebra. So diagnosis of a syndrome is like playing a puzzle. You have to put all the pieces together to get a final diagnosis. But this is not easy because the syndromes are in some cases very rare. And for this reason, we can help to use the so-called OMIM program. This is a database of human genes and genetic disorders. You can put in all the symptoms, and at the end you will get some suggestions which syndrome can be behind all these findings. And in that case, you can finish your puzzle with the last piece. However, in all these cases, you do not need only 3D ultrasound. You also have to use define the karyotype, and in most cases, you also need genetic testing. So let me conclude. 3D ultrasound with its different display options allows a detailed demonstration of abnormal findings of the fetal brain. 3D ultrasound enables a precise demonstration of asymmetry between the left and the right side of the fetal brain. In each case of any detected brain malformation, a detailed control of the entire fetus is mandatory. A syndrome must be suspected when several anomalies are found. And any detected fetal brain malformation requires fetal karyotyping, multi-gene panel testing or microarray analysis, or exome sequencing, and of course genetic counseling. But this is the most complicated part, because we can see several anatomic malformations, several anatomic abnormalities, but the question is always, what is the meaning of this malformation at the end when the baby is born? And we have, for example, a newborn of 3 or 5 months, and then we only can say we cannot predict all the prognosis in all these cases. Thank you very much for your attention.
Thank you very much, Eberhard. So the last speaker is me.
So Professor Pu is president of the Clinical Research Institute of Fetal Medicine in Osaka, Japan. She is professor of the Dubrovnik International University, Croatia, honorary professor, Pyrgov Russian National Research Medical University, Moscow, and adjunct professor of Wayne State University School of Medicine, Detroit, United States of America. Her main research is 3D sonar embryology, 3D neurosonography, and genetics. She has published many scientific papers and review articles in 3 ultrasound books. She got several awards. She is board member in several medical societies and vice president of the International Academy of Perinatal Medicine. Furthermore, she is in the editorial board of several international journals. She is not only a leading figure in 3D ultrasound, and particularly in neurosonography, but also an artist in scientific images. Professor Poo will give us now some interesting insights in neurogenetics. Ritsuko.
Thank you very much, Eberhard. This is my last talk, in this webinar, and probably everybody is already a little bit tired, but please be patient with the 20 minutes. Now, the brain development includes the neuronal proliferation, differentiation, migration, myelination, and many of the brain, the neurons are in the adult brain. However, most of the adult neurons are produced during the first and second trimesters, in the fetal period. And after that, the neuronal migration occur, and there are 2 migrations pattern, the radial migration and tangential migrations. And during the second trimester, mostly, the neuronal migration occurred. And meanwhile, the apoptosis occur, but this is not the bad meaning. This is a programmed cell death. It is necessary for the cells, the neurons, to adjust the number of the nerve cells to size of target. So the neuroimaging, should meet genetics, because the over 100 genes have been described encoding major pathway components mutated in isolated or syndromic hydrocephalic disorder. So many of the causes already that you learned from Vincenzo and also Eberhard, and many of the hydrocephalus has the causes of the hydrocephalus, and the many of the genes and many of pathway are related very deeply to the hydrocephalic disorder. And also here, it is one of the pathway, the mTOR pathway, and also the tubulin is the very important gene, and the mutation of those genes are very much related to the malformations of the cortical development. So MCD result from the diverse molecular disruption of the normal brain development and manifest as a wide array of the anatomical functional phenotypes. Now, this is the hydrocephalus. At 27 weeks of gestation, everybody agree that that this is the ventriculomegaly, very severe. That it is more than 2.7 centimeter, and hypoplastic corpus callosum. And here you can see that this is adducted thumb. Now we have the recent cases, the recent case with adducted thumb, and here the Professor Kuriak called that this is a neurological thumb. And here, this is the right and left, the both sides, the neurological thumb, adducted thumb. And this is the case of the L1 COM gene mutation. And this is X-linked hydrocephalus. of course, this is the X-linked recessive. The mother is a carrier and father is healthy, and then because of the X-linked recessive inheritance, the one of the son, the 50% of the son has been affected, the L1 syndrome. So actually, the L1 syndrome, already Karina had the lecture on the L1 syndrome, where L1-CAM is one of the cell adhesion and molecules belong to the immunoglobulin superfamily, and plays a critical roles in neuronal migration, axonal growth and guidance, and the fasciculation and synaptic plasticity in the central as well as the peripheral nervous system. So L1 syndrome is, not just the hydrocephalus. Now, this is from Professor Ito's paper, and this is a mice, but the L1 knockdown cells, you can see the difference between the normal and L1 syndrome. The green cells are here, the stops here during the migration. So the L1 syndrome is not a simple hydrocephalus and cannot be treated by shunting procedure. L1 syndrome is very severe, the prognosis, and L1 syndrome is one of the MCD and hydrocephalus. Now, this is the asymmetrical brain with mild ventriculomegaly. So what is happening? Here, this is the bigger hemisphere and the smaller hemisphere, and this is because of the AKT1 gene mutation mosaicism. But this is a somatic mutation, so in both brain hemispheres, AKT1 gene mutation was found as a mosaicism. However, from amniotic cell and maternal blood, paternal blood, all of them were negative. So this is a somatic mutation. And here, this is the big, the brain, the big head with a macrocephaly. And here you can see the abnormal sulcation here, and between the 18 and 20 weeks of gestation, the cortical malformation occurred, the polygyria. And the baby was born, at 21 weeks of gestation, termination of the pregnancy, and SNP microarray showed the mosaicism of the paternal UPD, uniparental disomy of 10q. And also, we did the exome sequencing, and P10 mutation occurred, and P10 is existing on the 10q. So this is our estimation, and most of our estimation, the sperm with a P10 mutation fertilized the egg, and then there was the mosaicism, of the LOH and non-LOH. So this is a schematic picture. One of the sperm had the P10 and fertilization, and then because of the paternal UPD derived from the somatic self-repair or somatic recombination, it is unknown. However, because of the paternal UPD mosaicism with P10 mutation, this very abnormal megalencephaly with cortical maldevelopment brain occurred. Now, this is another case, the big head with an asymmetrical brain. Here you can see hemimegalencephaly, and this is the the quantitative analysis of the asymmetry. The right brain is almost the double size of the left the hemisphere, And this is the overgrown vascularity, and this is the same at the same gestational age, the vascularity of the brain in normal case. But the left-hand side, you can see the overgrown vascularity, and this case had a tumor-like overgrowth skin. And exome sequencing, of the amniotic cell was negative. And also, the maternal-paternal blood was negative. However, after the autopsy, we take the brain lesion and skin lesion, and those show the PIK3CA gene mutation mosaicism. Now, the somatic mutation mosaicism occur because, the mutation occur during the mitosis. Therefore, there's no the mutation of the oocyte or spermatocyte, and the zygote has no mutation. Therefore, after that, somatic mutation occur during the mitosis. If the somatic mutation occurs, in early stage, maybe the baby has mosaicism, in the body. However, the somatic mutation occurs, later on, sometimes, it may be because of the environmental cause, sometimes, the local abnormality occurs inside of the brain. And this is very, the complex, the figure, but mTOR, the mammalian target of rapamycin, the mTOR signaling pathway integrates both intracellular and extracellular signals and served as a central regulator of cell metabolism, growth, proliferation, and survival. So, I showed several cases, related to the PIK3CA, AKT1, and PTEN, those are in the mTOR signaling pathway. So I told you that, the migration occurred during the pregnancy. However, can we predict that this brain at 20 weeks of gestation is going to the normal development or MCD? It is a challenging of the neuroimaging. So, we had several points or several factors or findings for the MCD detection in early second trimester. So first of all, the Sylvian fissure. The Sylvian fissure is, the growing, like this, and going to the Sylvian fissure, normal fissure, in late pregnancy, there is a closure of the fissure. And we did measure the Sylvian fissure angle, and then 25 weeks, almost 25 weeks, the rectangle structure of the Sylvian fissure, and this is the case of the abnormal MCD with chromosomal abnormality. And those cases, the lower part of the cases, that they had abnormal Sylvian fissure, and the Sylvian fissure is one of the signs for the prediction of the MCD. So more than 95% of the cases with the MCD, the Sylvian fissure angle were larger than the normal reference, and they have, the more than 30% of them had the genetic abnormality. Now, this case was referred with because of the IUGR, and here you can see, that this case, the 27, 29, 31 weeks of gestation, we did the neuroscan, and almost the rectangle. I said that, that is a 25 weeks of gestation. However, this case, 29 weeks. So, the 4 weeks delay of the Sylvian fissure. So this is the case of the microdeletion of the chromosome 13. And then, this case, she can work, and however, still, cannot speak yet, at the age of 1.5 years. And also, that this is the normal medullary veins at 25 weeks, and this is the premature, and this is one of the signs. And the other one, the abnormal circulation, this is a case at 19 weeks of gestation. There is a very abnormal asymmetrical brain. And here you can see the right side of the hemisphere, the right hemisphere, you can see the very abnormal gyration and circulation, but left hemisphere, there is no circulation. And amniocentesis, the TES, the exome sequencing was normal, and this baby was terminated at 21 weeks of gestation. And then we took the brain tissue for the exome sequencing, and then we had the abnormal, the OFD1 mutation mosaicism only in the part of the, the MCD part of the brain. Now, so that this is also the somatic mosaicism. Now, the irregular ventricular wall, already Eberhardt, Professor Meltz showed the periventricular nodular heterotopia, and this is one of the case, the periventricular nodular heterotopia. This is also the periventricular nodular heterotopia on the wall of the ventricular wall. And I said that, the radial migration and tangential migration in the migration disorder, migration of the neuronal cells. And here, the ventricular zone and the subventricular zone, and there is the excitatory neurons. And if the baby neurons cannot migrate, that they are remaining here, therefore, the hyperechoic VZ and SVZ. And this is the another one, the inhibitory neuron from the ganglionic eminence. They are migrating tangentially, and those are the normal ganglionic eminence, by ultrasound. And this is the case with a persistent ganglionic eminence. And this is from another group that the cavitation of the ganglionic eminence, and we had the case here, the ganglionic eminence, but that there was a cyst here, and this case was a case of the microencephaly, and because of the TUBA1A mutation. So here you can see at 19 weeks of gestation, the ganglionic eminence, and with a cavitation inside of the ganglionic eminence. This is the case, the after birth, TUBA1A mutation, and the very severe the neurological deficit after birth. The last three, the interhemispheric opening, during the 13 and 20 weeks of gestation, interhemispheric opening is opening, the closure first, but after that, the physiological change of the opening at 16 weeks, and then 18 weeks, the V shape, and going back to the Y shape. So sometimes that we have the gold sign, abnormal interhemispheric opening, and we had the 5 cases, and there are the 3 of them had the mutation of the single gene, and the 3 of them they deliver the baby the term, and so they have the neurological deficit after birth. So this case, the PPP2R1 alpha mutation on chromosome 19, and this is a baby, However, that she has to go to the blind score, from this year. Now, the posterior fossa, the many of the speakers already told about the Joubert syndrome, and Joubert syndrome has, the 36 subtypes, and each subtype has the responsible gene. And the Joubert syndrome is a genetic disorder of the cellular cilia, and not only brain disorder, but many organs disorders. And the very famous, the molar tooth sign, and I would like to show, just the case, in early pregnant case, at 12 weeks of gestation. Here you can see the diamond shape of the posterior fossa, and here this is 14 weeks of gestation and 16 weeks of gestation. This is not like a molar tooth sign, it is a little bit similar, but we cannot say that that this is the molar tooth sign, definitely. However, this case, bilateral renal multicyst occurred from 16 weeks of gestation, and we found that the CEP219 gene mutation, and this is a compound heterozygous mutation. Now, of course, the NGS is not perfect, and the cases with a negative NGS result, that we have this case, very abnormal posterior fossa, the very abnormal Barmian structure of the cerebellum, and this is the afterbirth, and here you can see, this is, you can put the focus on the right and left hand, the hands, and at the same time, the clench and unclench, if the right hand is unclenched, at the same time, left hand already also, the unclench the hand. So this might be the congenital mirror movement disorder, and we are waiting for the result of the sequencing. And this is the case of the 17 weeks, and there is the very abnormal tumor-like structure here. And 21 weeks of gestation, there is the very abnormal circulation. And this is the autopsy. And however, negative NGS result. So the both are perfect. The neither is perfect. The neuroimaging or genetics. Therefore, the fetal neuroimaging and the genetics are very tightly. They're corroborating each other. However, the fetal neuroimaging has greatly contributed to the elucidation of previously unknown fetal neurology. Thank you very much.
Ritsuko, thank you very much for this fascinating presentation. I think we are far away from 2D ultrasound, and it's not only 3D ultrasound, but it's even more. And I think prenatal diagnosis will be more and more complicated in the future, and you need a lot of experience to find the correct diagnosis. So I think we have now time for some questions or some comments. I can see here in this chat box, I can see one question. That question goes to Barbara, I think. The question was the case of de novo transportation at a transcerebellar diameter below the 50th centile or below the 50th centile.
So I already tried to answer. So at 22 weeks, it was just below the 50th centile. So it was within normal range. But what was striking that the baby was quite small. So the estimated fetal weight was on the 9th percentile. So this was the reason for a follow-up exam. But at 27 weeks, it was already at the 5th percentile. And later on, the growth slowed down. So it was below the 50th percentile from 27 weeks on.
Okay, now I will ask Anna, do you have any further questions from the chat box or from the question and answer box?
No, I don't. Just to see something came now, I think.
I can find one, and maybe for the Vincenzo, what if the anterior horn is dilated? Maybe for the ventriculomegaly, sometimes we see the anterior horn is dilated. The case is without anterior horn dilation. So maybe the Vincenzo, if you have any answer of this question. Vincenzo?
The ventricle assumes the shape of a drop. We have to think about on corpus callosum anterior. The dilatation only of the anterior horns may be the signs of neuronal migration proliferation disorder.
Thank you very much. So is there any other questions, Anna?
Yes, I send it to you. Which other CNS malformations will have molar tooth sign apart from Joubert's syndrome? This is a question from Umber Agarwal.
Would you please say it again?
Which other CNS malformations will have molar tooth sign apart from Joubert's syndrome?
Eberhard, you answer or?
Well, I think the Schubert syndrome is the most important one. Of course, you can have similar signs that are similar to a molar tooth, but it's rather difficult to differentiate. So I would be very careful with this sign.
Yeah, I think so. Yeah, actually, the molar tooth sign shows, the defect of the vermis of the cerebellum with the, the split of the brainstem. Therefore, sometimes, there is the defect of the vermis, and without the split of the brainstem. Sometimes they say that the Joubert syndrome, but maybe that we need the genetic confirmation. But actually, the genetic confirmation is sometimes very difficult, because exome sequencing, that shows the exome result. However, sometimes, Joubert syndrome has a mutation, not inside, the exome, but, a little bit far from exome, within the intron. So, no, this is not the imaging. But the genetic. However, anyway, the Joubert syndrome molar tooth sign is sometimes they're very difficult to the diagnosis. Is there any other?
Okay, I have one more.
Yeah.
What is the upper limit of anterior horn during 18 to 23rd weeks of gestation? Upper limit.
Yeah. Anterior horn, upper limit of the anterior horn. Is there anybody who can answer this question? I actually know that I don't measure the anterior horns, so that I cannot answer.
So I don't measure anterior horn.
So is anybody measuring the anterior horn, or do you think that that is important to measure the anterior horn? Eberhard?
Well, I think it should not exceed 8 millimeters, definitely.
Okay. I have one more. What's the prognosis for cavum vergae persistence at birth?
There is a maybe the cavus the verga is prominent at birth. So is there anybody answering this?
In my experience, is a normal variant without any clinical significance.
Vincenzo?
Yes, in my experience, it is a normal variant without any clinical significance.
Yeah.
So if we are looking in the literature, the cavum verga should be present after birth, and it can be closed after 3 years. If it persists after this age, maybe somebody has to control about the development of the brain, because we know that after 3 years of old, the connection between the axons go and develop further. So I can say that sometimes we see and we do not see the cavum septi pellucidity also in the prenatal ultrasound. Most of the studies say that if it's an isolated finding, it's not with worse outcome, but if we find some other abnormalities, then we have to check further. But till birth, we have to see, and also with 40 weeks of gestation, when we have performed the measurements of the corpus callosum, it should be present, the cavum septi pellucidate. Just the cavum verga has to be closed in the prenatal life. And as I read from the literature, it should be till the 3 years old babies.
So maybe at birth, if it is persisting, if there is the simple finding, it is not the adverse sign, right?
If it's persisting the cavum verga, the posterior part. Yeah. But cavum septi pellucidity is a normal finding. And if both of them are big, so one have to check, because maybe there are some migration disorder which we cannot diagnose prenatally.
Okay, thank you very much. Another question is it possible to see hypothalamus and pituitary gland on neurosonography? I think Reinhard Altmann can give some hints.
I think we have to wait a little bit until that structure will be fine displayed, and we have to work on that. It's a little bit too early, I think.
Thank you. The next question is, this is a complicated question. What would be the next tests if exome sequencing is negative despite recurrent CNS abnormalities?
Maybe that is for me?
I think so, yes.
The next test to the exome sequencing. Now, exome sequencing is negative. The exome sequencing, maybe, the target exome sequencing has several varieties. We did, the targeted exome sequencing with the 4,813 genes. However, recently, we are doing the 6,704 genes exome sequencing. However, of course, that this is exome sequencing. So if that is negative, you have to go to the full genome sequencing. And then the several cases I showed you today, that was, the somatic mutation mosaicism. That is inside of the brain only, the mutation occurs, only inside of the brain, not the whole body. Therefore, that there is no the negative data if we do the amniocentesis and full genome sequencing. Maybe the result is negative. Therefore, sometimes you have to think about somatic mutation inside of the brain. Is that the answer of you, Desai-san?
I think it's okay to answer.
Okay.
Another question that is what was raised, and I think you have to give the answer. Is SNP array preferred over CGH arrays in genetic testing, and why?
That is also, for me, right?
Yeah.
The SNP array is preferable, to CGH array because, the SNP microarray can detect LOH, loss of heterozygosity. And the CGH array, is only for the microdeletion and microduplication. However, SNP microarray detect not only the microdeletion, microduplication, but also they can the SNP microarray detects the LOH. And so, for example, as I showed you, the uniparental disomy, UPD, can be found by SNP microarray, not by array CGH.
Okay. The last question. Can you comment on the importance of tegmentovernian angle for the diagnosis of posterior fossa anomalies? I think this is a question to Barbara.
Yes, I think it's a very good measurement to evaluate the rotation of the vermis. If the rotation is quite high, so the angle is, I think, about 45 degrees, then you can assume it may be at any water malformation. If it's below, it may be a plaque cloud cyst. But I think it's quite difficult sometimes to measure it accurately because you have to have a very good beginning point. So I think it's not so easy to measure it very precisely.
Okay, thank you. So Anna, do you have any further questions or?
What values of HC do you use to diagnose microcephaly minus less than 2 or less than 3 SD with charts, within charts?
Well, I personally do not use SDs. I only use percentiles. And if it's below the 3rd percentile, I am defining this as microcephaly.
Okay.
I am using the standard deviation SD. Maybe in the United States, that they use the SD, and in Europe more, you prefer to use the percentile to the standard deviation. But in terms of the standard deviation, I use the -2 standard deviation.
But I think you also should look from outside to look at the fetal head, that you can see the shape. That is very important, that you know exactly where you have to measure. So another question is, which charts do you use for corpus callosum biometry? This is for Sunila.
I use the charts I developed myself because if you go to the literature, we have also conflicting data in the development of the charts. So available in the literature are the charts from Aheron. We have also some MRI studies. The MRI studies are describing a non-linear development of the corpus callosum as we do. But if you go to the Aheron, then they have linear growth of the corpus callosum. And this is a little bit, for me, a little bit strange because corpus callosum developed not only longitudinally, but also vertically and horizontally. So it's probably not the correct one. If you also see the recent publication, they try to confirm that they have a linear growth of the corpus callosum, but it's such a complex structure that I do not believe that is so. So our studies are more with the MRI, and all the other studies on ultrasound say that the corpus callosum is just linear. I am sure what I have done. That means that I have taken the median plane of the corpus callosum and I have measured just the hypoechoic area of this structure and all the 4 parts. And the other studies have just one plane for the thickness of the corpus callosum, and they do not decide which part was measured. Was the genome, was the rostrum, was the splenium? So that's why I use just the charts I have published.
Okay, thank you very much. There is, you showed precisely that you can have 3 measurements. You have the outer length, you have the inner length of the corpus callosum, and you have trace length.
Yeah.
So which one do you prefer?
I prefer the curved one because sometimes we have some part missing in the middle of the corpus callosum. So we have some pathological case where the body of the corpus callosum is missing. And so in these cases, I have difficulties of measuring the outer and the inner because I am not sure. So if I measure the curve of the 2 parts, then I know how long it is. It is important for me not only the measurements of the structure, but the structure that are present. Why? Because they have different accents. And these accents can be a challenge in our prenatal diagnosis. So we can say are missing the accents that are responsibility for the auditory fibers, or are missing the fibers that are for the motor function. And so maybe this is a future change in the consultation of the patients. But we have to work together. What is in corpus callosum is, first of all, we have to think in the same way, not just as a structure, but as a structure that have different functionality inside. So we have to measure the structure, we have to define the pathologies of the corpus callosum, and then to diagnose in the pathological cases if the splenium is missing. For example, we have some there are some literatures, some articles in the literature that these babies have problems with the eyes or with visualization of the objects. We have to see what is with the genome, with the truncus, and this will be a new method in consultation if we do not find something genetically.
So thank you very much. I think we have to come to the end, but there is one last question that is going to Barbara. What terminology would describe a vermis with normal superior inferior vermian diameter, normal AP diameter, but perimeter and surface area less than 5th centile?
So I think from a mathematical point of view, this is not possible, because if you have both measurements normal, then the area must be also normal. But sometimes there are some discrepancies. So I think the surface area is maybe the best to measure the vermis and to decide whether it's a vermin hypoplasia or not. And this was also suggested by the group of D who published the lobulation of the vermis 2019. They also suggested that the surface area may be the best.
So thank you very much, Barbara. I think we have arrived at the end of our webinar. We do hope that you enjoyed the lectures. It was a pleasure for us to communicate with you via internet and to share our experience with you. However, you can see neurosonography is not a simple field that you can perform with 2D ultrasound. You need a 3D ultrasound, because this is the only possibility to find out whether you have asymmetry in the brain. And this you can only define if you have the multiplanar view. You can correct the different planes. And also, you see, you need a lot of experience also in genetics, because many brain malformations are in association with gene defects. So last but not least, we would like to thank all speakers for their excellent and high quality presentation. I personally enjoyed it very much. We would also thank Anna and Jadranka for their preparation work of this webinar.
Thank you.
Finally, we do hope to see you again in the near future, however, in person and not via internet.
Yes, I hope so.
I only can say have a nice weekend and stay healthy. Ritsuko and Osim, do you want to say a few words at the end?
Yeah, actually, I enjoyed very much, this webinar, and this is very high quality, and every speaker had a very excellent presentation, and maybe that we would like to continue this webinar, in near future, and we can discuss much more, much further in the neuroimaging. And lastly, the Karina left, during the meeting, but that she said, she gives, her apology to all of the panelists and the participants because of her personal the matter. So thank you very much for attending this wonderful webinar, and maybe see you in near future. Asim, do you have any
last words? Go to Asim.
Just 2 sentences. I have no words to thank you. It was true scientific, inspiratory topic, well done within activities of International Academy of Perinatal Medicine. 2 weeks, 3 weeks ago, we discussed functional development of fetal brain. You outlined so nicely advances in the morphological study. Ritsuko is right and Meritz is right. We will continue in this way. There are many things to be said, in particular from our colleague neonatologist. So the next webinar will be recent advances in neonatology. We will see what our colleagues can tell us about the same person in different part of its life. One stress one experience which I've got listening and enjoying is that we can really have to make difference between ultrasound used by experts and on routine basis. Let's say fascinating new field of neurogenetics, which Risiko pioneered, plus that really person like she specialized in both genetics and 3D sonography can really fascinate and disseminate all of her genuine ideas into training systems. So more than ever, we need more education, and education is lifelong process. So there is no time in your life in which you can say, I am educated enough, no more. Yes, lifelong, and thank you very much.
Okay.
Thank you all and have a nice weekend and hope to see you soon.
Bye.
Thank
Bye.
Without, stay healthy, please.
Bye.
Yeah.
Of course.
you. You too.
Inshallah.
Sayonara.
Sayonara.
Bye-bye.
Bye.
Bye bye. Bye bye.